good. So I'm really happy to be here to celebrate Klaus's life and his scientific accomplishments. So I met Klaus in September of 1969 when he came as a graduate student to Harvard, and we shared an office together for I think 3 years. And I'd just like to say a few words about something that you might not know about Klaus, and this is something that I think Klaus might not have known about himself, because as you know, for a great scientist, Klaus had a terrible memory. And in fact, Dan once told me that at one time the primary function was to tell Klaus who his enemies were. That was true. In any case, when Klaus came to Harvard, he wasn't passionately interested in biology. He was interested in scattering theory. This is something that he wrote his master's thesis on. And scattering theory was very sexy in 1969 because of the recent invention of molecular beams. And in the course of his work, he had to calculate the vector coupling coefficient, the Krebs-Gordon or the Wigner's 2J symbols for large values of angular momentum. And in fact, a very famous Italian physicist called Tullio Reggi, who was famous for Reggi poles, which was something else that was sexy in 1969, and these were singularities of the scattering matrix as a function of complex angular momentum or something. Anyways, this guy conjectured some formulas for the classical limit of 3J symbols, but not for 6J symbols, but didn't derive them. So anyways, Klaus set out to find analytical expressions for these, and the way he did it was very clever. And he first showed that these coefficients satisfied a three-term recursion relation, which is a kind of a difference equation. And the continuum limit of this recurrence relation was a second-order differential equation, just like the Schrodinger equation. And all of you know that to get the classical limit of the Schrodinger equation, you have to do the WKB method. So Klaus set out to use the WKB method for a difference rather than a differential equation. And I remember that, once someone does something, in retrospect, you can look back at it and say, that's pretty obvious. But the thing that Klaus did was so clever that I never managed to understand how he got this equation. And about 10 years ago, I asked Klaus whether he remembered, and of course he didn't. But in any case, he was able to derive these very elegant expressions that were previously conjectured, and then he obtained new expressions for the 6J coefficient. So I was always very impressed by his mathematical ability and how really good he was at doing analytical mathematics. So I think I went on long enough, so let me introduce our first speaker, Wolfgang Baumeister from the Max Planck Institute of Biochemistry in Munich.
Well, thank you very much for the introduction. Thank you for inviting me to this symposium in honor of Klaus Schulten. Let me say I first met Klaus in the early 80s in Munich in the context of PhD exams in the physics department of the Technical University. We both had arrived in Munich at about the same time, 1980-81. Klaus at the physics department and I at the Max Planck Institute. We met quite often, over the next couple of decades, but we began to collaborate only much more recently. As a matter of fact, I think it started in a sort of an indirect way when Elizabeth Villa, who is in the audience, had been a graduate student with Klaus, joined my lab, and she contributed to many projects in the lab. She began to transform from a computational physicist, biologist, to an experimentalist in the course of the time. And then, more recently, when Till was recruited by Klaus explicitly for collaborative projects, and I must say this collaboration is still going on, and it's going on quite successfully. Now, my talk today is about cryo-electron tomography, which has been the main interest of my lab over the past, well, something like 25 years or so. Now, cryo-electron tomography is the application of tomographic data acquisition and of tomographic 3D reconstructions to frozen hydrated samples, whether it's molecules or cells. By doing so, it combines the power of high-resolution 3D imaging with the best structural preservation that is physically possible. Now, why do we want to do electron tomography? It enables us to do structural biology in situ. Now, I think it's fair to say that traditionally, structural biology takes an extreme reductionist approach. Cells are taken into pieces, the components are purified, and then they can be studied by any of the methods structural biology has at its disposal. I think it's also fair to say that in recent years, or maybe for more than a decade now, I think awareness has grown that very rarely biological functions are performed by individual molecules. They arise from the interaction between molecules in the context of the cells. So I think this is a strong enough reason to be able to look at them and the interaction networks in the context of unperturbed cells. The idea was certainly there. There was a remarkable paper published by Hart, who was at Roux laboratory in '68. The paper was entitled Electron Microscopy of Unstained Biological Material with Polytropic Montage. And as you read the abstract, it's clear that the polytropic montage is synonymous with tomography. And the last sentence of the abstract states, preliminary results indicate that one may thus study unstained or unshadowed biological material at high resolution. Now, so the vision was there, but I mean, for a vision to become reality, also the technology must be there. Now, this paper had very little impact. It was hardly ever cited, and the reason is that it was tomography, as it could be done at the time, was applied to freeze-dried cellular material. And as you looked, I mean, at the images shown in that paper, there were hardly any recognizable features. So then we began to work on tomography when it was possible to look at them embedded in ice. And of course the challenge was the radiation sensitivity of biological material embedded in ice. And to overcome, I mean, this problem, it's clear, I mean, there were two components. One was what people call dose fractionation, basically distributed the entire allowable electron dose over as many projections as possible. And the second component was a technical one. In order to minimize exposure, one had really to be able to automate completely the data acquisition. And that was, at the time, there were no motor-driven goniometers, no pretty lousy CCD cameras, and no real computer control of the microscopes. So that was quite a challenge at the time. I think it was, I think in this paper here in 92 that we were able to demonstrate that by automating the data acquisition, one can, I mean, keep the total electron dose, the cumulative electron dose, low enough to study ice-embedded specimens. Historically, this was the first cryo-electron tomogram ever published. This was, I mean, something as trivial as lipid vesicles embedded in ice. It was at that time a collaboration with Erich Sackmann in the physics department of the technical university. Now, I think in recent years there have been enormous advances in electron microscopy technology that led, as some people call it, to a resolution revolution. There are, let's say, a couple of components. I think one should not ignore, the progress that has been made in automating electron microscopes. So microscopes can operate now almost autonomously, and that means we can collect very large data sets of fairly consistent quality. The second major advance is on the detector side, where CCD cameras, which perform pretty lousy for 300 kilovolt electrons, have been replaced by direct electron detectors. They're also allowed to collect data in moving mode and to correct for beam-induced motions. And last but not least, because there are the face plates. Now, I mentioned that briefly because it's work from our lab, from Palestine Darnett in the lab. I think basically, as many of you will know, if you have a biological sample, molecule, or cell embedded in ice and take an image in focus, there's hardly any contrast. So one has to generate phase contrast, and that has been done for many years by defocusing. But the consequence of defocusing is a weird contrast transfer function with oscillations and zeros in the contrast transfer function, resulting in this type of ghostly image, requiring some image restoration, and again, that requires usually, taking images at different levels of defocus. So the hope was always to find something equivalent to discerning the faceplate as it is used in light microscopy, and that was made possible by the Volta faceplate developed by Palestine Darnett in the lab. Well, you see the difference. This is the 20S proteasome, and I will come back to that a couple of times in the course of my talk here. Conventionally imaged with the focus phase contrast, and this is the faceplate. You see, there's a dramatic difference in contrast here. There's no limitation in resolution. This is a 2.2 angstrom map of the 20S complex. Now, this is particularly important in the context of tomography, where the options for averaging are fewer than there are with single particle analysis, where you have, basically unlimited numbers of copies of identical structures. Now, this is also an early example, relatively early example of tomography we have done. This was a collaboration with Alistair Stevens' lab at the NIH of herpes simplex virus. Now, this is a pleomorphic virus, so it could not be done by single particle analysis, which relies on repetitive structures. I'm showing it here also because it is, I think, the first example of what we call subtomogram averaging. So if a tomogram contains in it, features which are repetitive, we can extract them computationally and we can average them. So locally we can improve contrast and therefore also resolution. So you see here in this case, the capsid is a repetitive icosahedral structure. It could be averaged and then it could be back into context. That is a principle that is now used all the time in tomographic studies. Now, for quite some time, our tomography was limited to small objects which could be imaged in total. So basically nothing that was thicker than half a micro, maybe 1 micrometer in thickness, can be imaged. For instance, here we have the clostridium cell drawn on a grid here, and only the periphery of the cell is thin enough to be transparent to the electron beam. The central part here surrounding the nucleus is far too thick to be transparent to the electron beam. So one has to apply some sort of thinning in order to be able to study the entire landscape of the cell. Now, in principle, what one can do is one can fractionate the cells, one can isolate organelles, and can study them by tomography. But our definition of structural homology is a relatively strict one. It should apply only to a scenario in which the cellular environment is preserved in its entirety, which means all organelles, all supramolecular assemblies are kept at their native place. Now, there are two options to achieve that. One is sectioning, cryo-sectioning, as shown here, but it suffers from fairly serious disadvantages. First of all, you see, It's sort of wavy, it makes really poor electrical and thermal contrast to the specimen support here. But the most serious problem is that as you physically section amorphous vitreous ice, there's massive compression of something like 30–40% or so. That is, of course, very disturbing. So we put our resources, human and financial, into the development of focused ion beam technology. So what we have here is a dual beam instrument. We have an SEM, which allows us to observe what we are doing and to observe, the sample in its entirety. Again, it's a big deuterium cell here sitting on an EM grid. And we have a second beam, which is the gallium beam, the focused ion beam, which we use to ablate material. I don't want to elaborate too much on that. I think Elizabeth will probably talk about focused ion beam quite a bit. So we can have two different scenarios. We can ablate material just from above and create, a gradient of varying thickness, so-called wedge or ramp here. But the geometry we use most of the time, we ablate material from above and from below and create a kind of a lamella which is supported only by the surrounding ice. To show the difference that it makes, I take some work that we are doing together with Chris Balchevsky from Case Western. We are interested in rod photoreceptor cells, more specifically in the rod outer segment here. That is what we could do before the age of focused ion beam milling here. About 10 years ago, you see, this is already the limit of thickness we can study. There are some regions which are flattened and which are transparent, but most of it is impossible to study by tomography. Now, this is what it looks now. This is after focused ion beam cutting lamella of a outer segment here. And you see, wonderfully preserved structures of these disks. Now, they are extremely rich in information, but retrieving that information is not trivial. So what I've done here is at the terminal ends, they have done some averaging. Matthias Böge has improved that quite a bit in recent days. So this is the so-called molecule peripherin, which enforces these extreme curvatures at the end of these disk membranes here and maintains this curvature. But what we are really interested in is the supramolecular organization of rhodopsin molecules inside these membranes, and that's of course challenging. Rhodopsin is a relatively small molecule, and the density difference between lipid and rhodopsin and protein is relatively small, so it's a major challenge in terms of image processing. So we have to optimize the algorithms for the detection of rhodopsin and combine some sort of cluster analysis and detection algorithms with subtomogram averaging. But I think we are making pretty good progress on that one, and I think another 6 months or so we will be able to see what the organization of rhodopsin is and what the interactions are. Now, all these methods can be applied only to cells, individual cells grown on grids, or to cells deposited on grids. If we have a tissue, or if we have a multicellular organism, such as a C. elegans, then we need a different approach that is illustrated in this movie here. We cannot ablate material sort of horizontally. We have to cut vertically deep inside into the structure, and then we have to manipulate the block of ice. I think maybe I rerun movie. Okay, so we cut something vertically, then we have a gripper here, then we can free that block of ice. We have to transfer it to another holder to thin it further in order to study. But the technique is working now quite reliably, I have to say, and I think we can obtain decent resolution with blocks cut in this way in ice, and we can target essentially any volume in a multicellular organism or in a tissue. Now, I'll illustrate a bit what we can do with some work done with chlamydomonas. That is work done by Van Engel in the lab. So we have here a chlamydomonas cell embedded here in this lamina here. Now, Chlamydomonas has a relatively well-defined topology in terms of organelles in the cell, so we can zoom in on any part of the cell here. So we focus here on the Golgi apparatus. So this is the nuclear envelope. This is the nuclear pore complex here. See, the nuclear envelope is studied on the outside with ribosomes. We have a part of endoplasmic reticulum. We have vesicles like the Koch vesicles, which are intracellular delivery devices. Now, we can then cut out any volume from those tomograms, and we can do subtomogram averaging. This is work done in collaboration with John Briggs. So this is a Koch vesicle here, or part of that of the Koch coat here, at a decent resolution. The nice thing is, unlike reconstituted Koch vesicles, which have been studied at slightly higher resolution, in this case we have everything, including the cargo, and we also maintain the relationship to the topology of the Golgi apparatus. We can focus on the nuclear pore complex and averaging a relatively small number of nuclear pore complexes work done together with Martin Beck at the MPL. We obtain the decent structure of the nuclear pore complex in situ. Ribosomes are abundant, and we can easily pick a fair number of them, in this case a few thousand, and obtain a decent resolution by subtomogram averaging, in this case 8.5 angstrom, and this is without pushing the envelope. The interesting thing is that we can, of course, then focus on ribosomes in the chloroplast here. There are fewer, and with only, well, you see some of the ribosomes here in the chloroplast. Now, based on only something like 200 or 300 ribosomes here from a single chloroplast, we can again obtain a decent structure. Of course, resolution is a bit lower here, but the advantage is that here we have the purity by subtomogram averaging we could never achieve by chemical means. And there are interesting findings showing the discovery potential of the method. For instance, here we see a density well defined, which you never find if you isolate the ribosomes from the chloroplast, because it's very weakly attached. And of course, the challenge is that is going on. Stefan Pepper and the lab is working on that to identify what the corresponding proteins are. We can focus on ribosomes sitting here on the translocon, in this case of the nuclear envelope. This is work that was done by, again, Stefan Pfeffer, together with Friedrich Förster here, doing, averaging over the R-associated ribosomes here. They obtained a pretty good resolution, for instance, here of the translocon, very clearly visible alpha helices. Now, another series of examples comes from the work done by Julia Mohammed in the lab. So she focused on the periphery of the nucleus in the HeLa cell. So this is a HeLa cell, this is the nuclear region in the HeLa cell, very faintly visible. The lamella she cut at the periphery of this HeLa cell. And these are now the structures as we proceed from inside the nucleus to the outside. So here we are in the nucleus, and you see these chains of nucleosomes here. Techniques to develop decent segmentation algorithms to obtain deeper insights into the higher order structure of chromatin here. You see very clearly here the meshwork of the nuclear lamina as we approach the nuclear envelope. This is on the nuclear side, a single nuclear pore complex. This is the same one a little bit further out on the cytoplasmic side. And here we are now on the outer surface of the nuclear envelope. And you see here these rows of ribosomes forming polysomes, for instance. Now, Julia has done quite a bit of segmentation and averaging. I think just one example here, that is the nuclear pore complex. Now, this is the difference between with the faceplate and without the faceplate. It's not the highest resolution that has been achieved, but it's remarkable in that it is the result of averaging over a very limited, less than half a dozen of nuclear pore complexes. And this is important because I think it would be important to improve primary data quality further to the level that we can see a lot of detail in individual nuclear pore complexes, because there's a lot of variation, for instance, in diameter of the nuclear pore complex. It varies something like 13%, alone in this data set here. This is a synopsis of all the findings of UVA of the nuclear pore complex. Again, in the background, the chromatin, the meshwork of the nuclear lamina, then here the polyzone sitting on the outside of the nuclear envelope. Now, in the next few minutes, I will show you some work first, that is single particle work. But I need to do it for two reasons. First of all, I need to do it to introduce two players. I will discuss a little bit further then in the context of tomographic studies. And the second reason is that in one case, it involves quite a close collaboration with Klaus Schulten's lab, especially with Attila. Now, I think this is a very simplistic view of the eukaryotic protein degradation pathway. It's simplistic in the view that it shows only the executive end of the pathway. So proteins destined for destruction are marked for destruction by the attachment of ubiquitin. They are recognized by this molecular machine, the 26S proteasome, which degrades them in an ATP-dependent manner, generating oligopeptides which are typically 8 to 12 residues long. They are very little used for the cell. They need to be further degraded, and the first step is done by molecule 6 megadalton tripeptidyl peptidase, cleaving off tripeptides, and then they are degraded further by an array of aminopeptidases. So first, very briefly, tripeptidyl peptidase, that is a structure we have done several years ago by using a hybrid approach. So we determined the structure of the building block, which is a dimer here by X-ray crystallography, and that was then docked into the EM density map of the entire complex. So this is what we call a turboatomic structure. I don't need to go into the details. Basically, it is two strands of linear polymers built from this generic block here. Now, I have to elaborate a bit more on the 26S proteasome, also because that is where with Till and Klaus we collaborated quite a bit in recent years. First, this is the structure of the human 26S proteasome, now at a resolution of 3.9 angstrom. I don't want to go into much detail. It may suffice to say that the red part is the core particle, or 20S proteasome. That is where protein degradation takes place. It takes place in a cavity deep inside this complex here, which is sequestered, therefore, by the cellular environment and protects the cellular environment. Now, in order to become functional in the cell, it has to associate with what is referred to as a regulatory particle here. There can be one or two regulatory particles, and their role is to prepare substrates for degradation. They recognize ubiquitinated proteins, they deubiquitinate them, they unfold them through the action of this AAA ATPase module here, that also assists them in their translocation into the core complex. It is this regulatory particle which also controls a gate that gives access to the interior where the degradation takes place. Now, that was the basic organization. In recent years, we have seen that in its functional cycle, this molecule undergoes, especially the regulatory particle, very large-scale conformational changes. S1 was the ground state in which it's not actively engaged with substrate, and S2 is what we call the commitment state. And the process of binding substrates is initiated, but it's not yet processed, but it becomes irreversible, and S3 and S4 are two states in which the processing takes place. Only S3 is a state in which the gate to the core complex is open. So you see here we have large shifts and we have large rotations of entire ensembles of subunits here. They serve basically to bring down the substrate from the receptors initially to the mouth of the AAA ATPase, to position the resident ubiquitinase such that it can cleave off ubiquitin, which cannot be unfolded prior to unfolding, and also it controls gate opening. So I think the very simple message here is there are very large conformational changes during the functional cycle. Now we move back to tomography and in situ. We studied first, that was before really our focused ion beam was fully functional. We started hippocampal neurons grown directly on the EM grids. So you see here the hippocampal neurons, these are the grid bars here. Now, as you zoom in, so this was unthinned. I mean, these cells have many protrusions which are relatively thin. So they are sufficiently transparent, but not very thin. So a bit more than half a micrometer or so. So these are the raw data here. So the image looks at a glance very non-spectacular. But as you zoom in, for instance, on this guy here, these are slices now to an individual 26S protein. So you recognize basically the feature, the top complex, the two regulatory particles, and so on. So these are completely without any post-processing. Now, of course, you recognize the particles only if they are in a favorable orientation, basically with the long axis of the molecule perpendicular to the beam, but they, of course, occur in all possible orientations, so we have to perform a search with the template, for example, to some extent, but at very low resolution, I mean the structure here, and then we find particles in all possible orientations here. We can generate class averages, and we can then produce 3D reconstructions of the particles in the cell. That is the global average here. Now, in the global average, you see already that one regulatory particle appears fainter than the other one here. As you do calculate the variance map, you see the main variance is the presence or absence of one regulatory particle, and then you can make here a difference map. So you see we have, I mean, only about roughly 1/3 of the particles has two regulatory particles. The majority, more than 70% or so, has a single regulatory particle. Now we have distinct variances also within the regulatory particle, which we can separate again through image classification. And given the magnitude of the conformational changes, we can correlate one structure here with the substrate processing state and one with ground state. So in this particular, under these experimental conditions, roughly 80% of the particles are basically sitting there and waiting for substrate, and only 20% or so are actively working on substrate. Now the nice thing is that we can keep the coordinates where the molecules were found in the cell. And so we can map them back, and we can say where a particle is single cap or double cap, or whether one is in the ground state or in the substrate commitment state. Now, I said that TPP2 is a molecule that works downstream of the 26S protein. Now, the problem with TPP2 is it is a very low abundance protein. Roughly the molar ratio of TPP2 to 26S proteins is something like 1:40. So they are very rare. And you see them here. Here you see very clearly, again, in the favorable orientation, you see the characteristics of TPP2 in the raw data. But in order to get a decent structure, we have to take, well, something like 70 tomograms, resulting in something like 93 particles here, and from that we could calculate a map at something like 20 angstrom resolution. So about the resolution we had 10 years ago with a single particle. But again, the nice thing is we can see now is their co-localization of 26S proteasomes and TPP2. And we can show by statistical analysis gains, so they form what we call degradation centers in the cell. Now, we look at 26S proteasomes, we go back to chlamydomonas in this case. Now, roughly half of all the 26S proteasomes is in the cytoplasm, And those in the cytoplasm occur essentially everywhere in the cell, but they're usually in close vicinity, forming, again, what we call degradation centers in the vicinity of the ER. So that is, in a way, the visualization of the air-like pathway in which disordered proteins from the ER have to be retro-transported into the cytosol in order to be degraded there. The situation in the nucleus, where the other half of the proteasomes is located, is very different. Basically, we see here, when we plot the distance of the 26 particles to the nuclear envelope, we see two peaks here, and I will elaborate on that a little bit further, but essentially all the proteasomes are in close vicinity to the nuclear envelope. And so we can play the same game again. And here you see the raw data. Now the data quality is already quite a bit better. So you see here on the original unprocessed data, you can already recognize the features of the 26 proteasome in relation to nuclear pore complex and here to the nuclear envelope. So this is again the global average here, and we can do, of course, the classification. Now, when we study those close to the nuclear envelope, then we find two populations of 26 such proteasomes. We find one population shown here in red that is relatively close, or very close, to the nuclear envelope. As a matter of fact, it's in physical contact with the nuclear envelope, but it's a little bit further away from, the eightfold or from the center of the nuclear pore complex. Now, this population here in yellow is a population that is a little bit further away from the plane of the membrane, but it's closer to the center of the nuclear pore complex. And again, we can map them back here. So you see here in yellow those which are further away, and we can show that they interact with the nuclear basket. And we have in red those which are in physical contact with the inner membrane of the nuclear envelope. As we map them back to individual nuclear pore complexes, the occupancy, there are essentially 8 identical sites because of the eightfold symmetry of the nuclear pore complex. In this case, 7 of the 8 sites are occupied, in this case only 6, and some have fewer. And you see also, when you plot the positions here, that you see here in this blue one, in this case here, they are not sitting on an ideal circle here. That reflects basically the flexibility of the nuclear basket. So when we do that for the entire data set, we can again discriminate between single and double-capped nuclear 26 proteasomes. We can classify those in the ground state and in the substrate processing state, based on their conformation, and we can say which ones are free, not interacting with any cellular structure, which ones are basket tethered, and which ones are membrane tethered. So in the cytoplasm, none of them is bound to distinct membranes or other structures, whereas in the nucleus, roughly half of them are directly interacting either with the nuclear complex or with the nuclear envelope. Now we can map them back again. So this is the homogram again. This is the mitochondrion here. You will see very nicely on the mitochondrion bases sitting here in yellow. These are the nuclear pore complexes. All those are the 26 proteasomes, those in the cytoplasm and in the nucleus. The single cat in a brighter color than the double cat. We can say which ones, the green ones are in the ground state, the other ones, the blue ones, are in the substrate processing state, and we can also classify them according to their interactions. So, the summary of this part here is, yes, we have 2 different interactions. One with the nuclear basket here, probably involved in surveillance of import and export, and one population bound to the membrane, and probably involved in maintaining the compositional difference between the outer membrane and the inner membrane of the nuclear envelope. I think in the last part of my talk, I will tell you about a project that is called Toxic Protein Degradation in Neurodegeneration, or TOPA. There's a collaboration between 3 departments of our institute, Ulrich, who is interested in how do aggregates affect proteostasis networks. Can we use cellular defense against aggregation? My colleague Matthias Mann, a mass spectrometry expert and proteomics person. What is the composition of the aggregates and how does the cellular proteome change in response to toxic aggregation? Our interest is what is the in situ structure of the aggregate and how do the aggregates interact with the cellular environment. Now that requires a relatively complex workflow. There's of course vitrification, always the first step. And since the cellular landscape is large compared to the field of view of the tomogram, we have to identify and locate the aggregates in the context of the cell. Then we can transfer the coordinates to the focus ion beam and cut the lamella in the right region of the cell, and then we have to transfer it to the microscope. Now that is the process in reality. So this is a grid with HeLa cells growing here on the grid, and the HeLa cells express Huntington inclusions. So it's a really long polyfluoride expressed in these cells. Now so all cells that show, the fluorescent label here carry Huntington inclusion, so we can focus on this one. This is an isolated cell, and we can observe it in the UV instrument. We can cut the lamella here. We can confirm that the lamella includes the Huntington inclusion here, and then we can take the tomogram. Now this is the tomogram of a Huntington aggregate, not in the HeLa cell, but in the primary neuron. We have grown directly on the grid here. The quality of the tomograms is such that automated segmentation can easily be applied. Fibrils are only something like 3 to 4 nanometers in diameter, but we have very clear and defined density. Now what you see invariably in all aggregates here, they interact with the endoplasmic reticulum. The endoplasmic reticulum is shown here in red, and it's more or less destroyed here at the periphery of these inclusions. Whenever the fibers touch membranes of the ER, the curvature of the membranes changes in a dramatic way, and that leads to disruption of the ER. And the consequence of that is then the liberation of all the ribosomes bound to the endoplasmic reticulum. The remarkable feature here is that even though there's a lot of space in this aggregate, the ribosomes and also all other macromolecular complexes remain at the periphery. They don't inhabit here this particular neurotoxic aggregate. Now, here we zoom into the network a little bit and show, first the segmentation, and then we can do quite a bit of quantitative measurements here. So you see the radial architecture of these Huntington aggregates. Now we can do a couple of measurements here. We can measure, of course, the length of the fibrils. We can compare it. What we have done here is in order to facilitate finding the aggregates, we have labeled some of them with GFP, and we have done it also in absence of GFP, and we want to see whether the GFP alters the supramolecular organization of the network, and it does to some extent. As you see here, basically the excluded volume by the fibrils changes quite a bit in the presence or absence of GFP. It's less densely packed in the presence of GFP. The persistence length, as a measure of the stiffness of the fibrils, is about the same. We see here that directly this is the fibril network in the absence of GFP, and this is in the presence of GFP, and the little dots you see here on the surface of these fibrils, they represent the GFP molecules. Now, there's a second type of aggregate now that's it's a model system. It contains a synthetic glycine alanine repeat, and it mimics or recapitulates the symptoms or the phenotype of ALS. It forms again aggregates in cells, and we can study them using the same approach. At a glance, it looks like these aggregates formed by the polyglycine alanine repeat protein here looks relatively similar. The volume that is occupied is about the same, something like 4 or 5% of the local volume. Now, as we do the reconstruction and the segmentation, we see these are not figures, however, they are ribbons or they are sheets, polymorphous sheets, very often with sort of these bifurcations here. But the main difference is that in between all these sheets here, you have a dense population of molecules, and you can extract all the particles computationally and then do averaging and classification, and that is the outcome of that analysis here. So we have basically the main inhabitants of that aggregate are indeed protozoans. So we have a local concentration of about 30-fold compared to the other cellular volumes. It's almost exclusively 26 protozoans that are trapped and inside these aggregates here. They obviously deplete the remainder of the cell from an essential component of its quality control system. We find, of course, ribosomes, but ribosomes are, again, they are only at the periphery, and strict, which is the main eukaryotic chaperone and what's downstream of the ribosome is also more or less at the periphery. Now, given the abundance of 26 protozoans, we can do fairly deep classification and averaging, and because of that, now we are roughly in the 1 nanometer range of resolution in situ here. So in green, you see in two different orientations, two ground states. There are slight variations, which are highlighted here, but I don't want to go into detail here. In blue, you have the particles which, according to their conformation, are in the substrate processing state. Invariably, they have an extra mass here, which is shown in this purple color, which we interpret as substrate. And you'll see now we can map them back again to the original tomograms. And you see the blue guys, I mean those which are in the substrate processing state, invariably are in direct physical contact with these ribbons or sheets here. Now, we can go a little bit further here and do a focus classification of the purple regions and see which part of the 26 protozone they interact. They all interact with RPN1, which I mean is a third, if you like, receptor. I mean it interacts with shuttling ubiquity receptors here. So, and I think there's still room for improvement. I discussed it a little bit with Phil during the lunch break. I think there's a pretty good chance that we push that to 6 or 7 angstroms in the future. So that brings me to the end. So that is the group of people involved in the work, I mean past and present. And again, I want to use this opportunity to acknowledge the collaboration with Till and Klaus. But thank you for your attention.
Any questions? I guess.
You know, you studied almost all the degradation systems. You had a very low resolution picture also of the faculty, and you have now these two. Do you understand the overall strategy for this degradation? I'm sort of surprised that it's sitting so close to the nuclear core. And then where is the faculty? I thought that that's sort of devouring the larger organelles. It's becoming effective. Do you understand the overall strategy?
Well, I mean, somewhat surprisingly, but there's work by others, including my colleague Wolfgang. I mean, the nucleus is a major protein degradation compartment in the cell. That is perhaps somewhat surprising, but it's quite well established. And I think that it's also well established that there's a lot of transport of protozoans into and out of the nucleus. So there's heavy traffic in that aspect. What is completely new is the very specific interaction of the nuclear protozoans with the nuclear pore complex, and two distinct sites. That is something that needs to be clarified further. We are currently in the process of identifying the adapters that are involved in that. That was completely unexpected.
All right, well, let's thank our speaker again. And our next speaker is Benoit Roux of the University of Szabo. He was actually working on his farm in second
Yes, so these are some remarks. It's a great honor to be here and great pleasure, and I have a few slides that are partly anecdotal. So this is a pointer still? Yes, this is it. It's good. This is the first time I met Klaus in 1993. It was in Strasbourg Oberlin conference that George Whiff had organized. I don't think you were there then. Don't think so. Stephen Fisher, Martin Karplus, this is Roland Stote, Marcy, this is Klaus, and this is me next to him. In fact, he was sort of my new buddy at the time. That's how we were together. Jeremy Smith, there's Misha, and D.E. McCammon. I believe this is Leonard Nielsen, not completely sure, Annick Jaguar. And so this was the first time I met Klaus. And then he invited me, I think, a year later to give a talk here in Urbana. And we were in touch over the years. This was 93. I was assistant professor in Montreal. And in 2004, there was the Nobel Prize on the potassium channel and on the aquaporin. Basically, there was Rob McKinnon and Peter Agri. And it turned out that actually we had done simulation of the potassium channel. Klaus had done good simulations of the aquaporin, and so as soon as the Nobel Prize was announced, I asked Klaus if he wanted to write a review with me. This was supposed to be a perspective published in Nature, which they asked us to write, and then finally they sort of ditched us, so it ended up in Structure. I don't know what happened. They seemed to be totally enthused, and then they sort of didn't want to do it anymore, so we sent it to Structure. Actually, when I submitted to Structure, I did not change the letter N to the Nature editor. I saw it afterward, actually. But they published it anyway. So. This was our first paper together, but then when I moved to the Midwest in 2005 or 6-ish, something like that, the papers started to accelerate because, Chicago and Urbana is not that far. So in the end, this is the last paper, number 12, got accepted yesterday. This is with Chris Schippo, also my other partner in crime, and spearheaded by Brian Raddack. This is the constant pH Monte Carlo method that finally accepted But so sort of the major paper we did was like, for example, this paper that was done by Fatemeh Khalili, who's at UIC in Chicago. And this was the calculation of the gating charge of the voltage sensor in the potassium channel. It's actually the first time that the gating charge of the channel was calculated with all atom and D. So it's actually a pretty highly cited paper. It's not super old. This is the first time that we implemented the Drude force field in AMD. This is the paper. That's also, I think Sharon handled that paper. It was like physical chemistry letters. Let's see. There was also a paper on the multiple copy methods with, like replica exchange, string method, all that stuff. And so this was published not a long, long time ago. Wei Jiang from Ardon was sort of the leader of that. But you can see many of the people, J.C. Kumar, so lots of people. And a lot of the most modern methodology of NAMD and many of the last calculations that Klaus described even at this keynote address by the school said they actually relied a lot on this multiple copy because they use a lot of string method and stuff at Eduardo Peroso, where they actually resolved the structure of a voltage sensor phosphatase, and it was fairly low resolution. So this was one of the first application, pretty high profile application of the XMDFF. So this is the MDFF, but the version for X-ray crystallography. And they published another methodological paper. I'm not all that. I do anything on that, but the contention is that this direct space of refinement of the density is actually sort of more robustly stable when you have low resolution data. It's kind of very encouraging, and of course that actually for something as pH. So I'm going to talk about a few topics, sort of maybe in the Schulten spirit of the last few with a few topics. The first one is the kinases. The system I started to work on many years ago, these tyrosine kinases, and not only the name of the people involved should be there if I'm bad at so Yilin Meng and Nikolai Fraser did a lot of that work. So the kinase is an enzyme, and it has a catalytic unit here. The domain is the binding site. It binds ATP and it transfers a phosphate ATP to another protein. But its point, of course, is not to burn ATP. Its point is to phosphorylate the proteins. And this is a biological signal. So this is supposed to be the down-regulated inactive state. And this is one possible active state. There's several. Essentially, anytime you leave the inactive state, you potentially could become active. So this is one that was crystallized, but there are several of them. And if you remove these SH2, SH3 domains, these are the regulatory domains, if you remove them, typically the catalytic domain on its own is constitutively active. And so when I started to work on that many years ago, the idea was like, well, maybe as soon as you remove this SH2, SH3, the protein just undergoes conformational change and becomes active. And so we wanted to see that, so this would be like, okay, when you have just the catalytic domain alone, does it want to become active? So what's the equilibrium for that? And then you would like to compare this to this kind of activation reaction when you have the regulatory domain in the inactive state, and perhaps the same reaction when you have conformational change when you have the regulatory domain in the active sort of state. And so, of course, the first thing you would like to do is, this is a large conformational change. It looks easy on the cartoon like that, but it's actually, there's a loop, it moves, it's in V, excess rotates, it's a little complicated. And this was one of our first application of the string method. So the string method is a multiple. This actually originates from work that Ron Elber did when he was postdoc, when I was a student. So if you have many copies of the system, and some people call that a chain of states, and you try to go from this first system to this last system, and everybody sort of had to resemble a bit their neighbors. And so the way we changed that a little bit is that rather than using the proximity to the neighbors as a kind of a geometrical restraint, what we do is we actually release these guys many times. So these are red trajectories, we call that the swarm trajectories. We let them diffuse randomly, and we take the mean drift out of that to try to pick up what is the direction of the stream of systems in this space. And then you kind of update that little by little. And this is basically that algorithm applied to this little toy problem, but you can see that actually it does refine the pathway towards the correct saddle point. So the nice thing about the swarm like this is that it's extremely easy to parallelize because you just have many copies. So when you run that, for example, on blue gene, you could have, we typically have like tens of copies of the system, and every swarm is a copy. You would have like 100 swarms, so you get like, 10 times 100, you could have thousands and thousands of copies of the system sort of blasting away on the blue gene and recollecting the data. So that's like pretty effective to do that. And so that paper was published in the special issue of Jason Scale for Klaus. And Nikolai is the first author. So we did like all the pathway of the kinase in the two states. We did actually the catalytic domain alone. We did the downregulated and active state too. And these pathways look a bit like that. You know, it's actually this a more ancient pathway. But more interestingly, also we can use the pathway as a seed to start umbrella sampling. Because once you have a nice pathway, you just have to expand a little bit around the pathway to sample. And so we use that to do umbrella sampling. So this umbrella sampling has a free energy surface of the kinetic domain alone. This is inactive, and this is sort of active. And you can see this is blue, so that's lower in free energy, and then when you get there, it's not super high, but it's higher, you know. And this motion is the motion of the loop, and this motion is the motion of the helix, the alpha C helix, we call it. And so it's pretty clear from the surface, although we don't inspect that, but we know from other calculation, actually, the string wanted to basically move the loop first and then rotate the helix. And the helix is something more chunky to move, whereas the loop is something more floppy, so it's easier to move the loop at the beginning. So you can see that you can visit the active light state, but it's a higher free energy. So this did not quite, initially this did not jive with the idea that the kinetic domain alone is constitutively active. But when you start to think that, well, if you bind ATP, people know that these kinases actually will autophosphorylate, in the sense that a kinase will collide with another kinase and phosphorylate the other kinase. And so in solution, they try to cross-phosphorylate each other like that. And so if you make a kinetic model with this state, and this state, and you can phosphorylate them, this will actually rapidly become constitutively active, because it's, you have like enough probability to visit that state. Now what happens is you have all the regulatory domain in place with the down-regulated state. You can see now you're stuck down there, and the free energy to go visit that kind of active light state is very, very, very high. And so that means now you would be really locked in an inactive state. So that's kind of nice to know that. This calculation was done about 3 years ago. It was aim 2 of my first NIH grant on the kinase in 2002. So we were a bit over-ambitious, I think, in 2002, because it took only about 13 years to actually get it. So if you do the active light state, interestingly, you stabilize the active state. And so, this is, we actually have done also calculation when you phosphorylate this loop, because this tyrosine 416 can get phosphorylated, and it's the same. So, it's, the free energy surface is really modified by these regulatory domains. It's very interesting to see that. And so, many of these calculations were done with the multiple copy aspect of NAMD for umbrella sampling, where you do a change between the windows. And so, that's also something that was done like that. On ion channels, another story that's ongoing. So it's about inactivation. Inactive channels conduct ions, but they, and I used to work a lot on ion permeation, but the more interesting biological aspect is not so much just the permeation, that's just like the basic raw signal. The real interesting thing about channels is their regulation, right? When they conduct, when they don't conduct. And so, channels like this is a potassium channel, the KCSA channel. If you give it an impulse, a stimulus, and this particular channel is stimulated by a drop in pH, so you go from pH 8 to pH 3. This is a stimulus. The population channel will conduct. So this is a population channel conduct. But overall, statistically, they tend to stop conducting, even though the stimulus is there. So that process where they lose their ability to conduct is called inactivation. And it's somewhat mysterious. It's been known for many years. And this is EPR experiment that Eduardo Peroso shows that actually when you drop the pH like that, the bottom gate really is open and remains open as function of pH. So whatever it is that makes these channels stop conducting, it's not because the bottom gate is closing. It is because the filter or something else is stopping to conduct. And so we try to understand that better, but schematically, this is the view that people have, and we certainly have that. Like the channel can be closed, and the filter can be poised to conduct. You open the gate by some stimulus, the channel conducts, and then you sort of decay into a channel that still has the gate open, but somehow the filter is unable to conduct. And then the only way you can sort of recover that is remove the stimulus, and this will sort of go like that. And so experimentally, this is what they do. They will give us a channel, you give a stimulus, you open it, it conducts. After a while, it has inactivated, you remove the stimulus, you close it, you wait some time, you're ready to go again. Now, the time you wait is the recovery period. And it's interesting to know how fast channels take to recover. So it turns out that if you measure the recovery period like that for KCSA, it's extremely long. It's like several seconds. And we heard this morning like about 10 amino acids per second to synthesize a bacteria. Well, KCSA is 160 amino acids, so you're talking about something that is basically taking as much time as bacteria to synthesize KCSA. It's like 10 seconds. And so it's a conformational change, supposedly, but it's so slow. What could it be? There was a structure from Rod McKinnon in 2001 that was the conductive form, but he got another structure that is called, these are PDB-ID. 1K4C is the one that is at high potassium. This is the one that is conductive. But there's also another structure you got at low potassium. That's the 1K4D. And it's sort of pinched here. So there's no, the ions are not in the core, they can't quite go through. This is the pinch. If you remove the ion and superimpose them, you can see the pinch is a little bit narrower. However, the difference in RMSD for the atoms of the field is about 0.7 angstrom RMS. It's a little shocking to imagine that 0.7 angstrom RMS would present such a challenge that the protein has to take 10 seconds to overcome that conformational changes. Doesn't really make sense, you know. So for many years, I thought this structure has nothing to do with that inactivation. I don't think I was the only one. I think even in the paper from McKinnon in 2001, he stays away from saying this is an inactivated pore. And to this day, I think he has never really said it was. So what we started to simulate that. This was actually done on Anton because if people give you a lot of Anton downtime, what can you do with other than just throw it there and simulate the long time? So we were kind of curious. It was a bit of a fishing expedition. And so we simulated this with 25 microseconds. It was the channel in the pinched state. And I was sure it would sort of become conductive because I thought this is just like a minor distortion. It will just leave there and become conductive again. It did not. It stayed pinched for 24 microseconds. And I said, I can't believe this. This makes no sense. You know, this is like 0.7 angstrom RMS. Why is it doesn't pop into the other conformation? And so Jared, my student, he observed that actually there are water molecules bind behind the filter, but they bound there. They are in the crystal structure. Very often when people build their MD system, just throw away the crystallographic water because you know, you're going to soak it with water anyway, so that's not a big deal. But you know, when water is actually bound in the structure, it could be a little bit more tricky for these waters to return there from the simulation. So here you have been kind of careful. You have done that very carefully. And you can see these waters are not so innocuous because if you try to replace the filter, first of all, there's 3 waters like that when you're inactivated, but the crystal structure of the activated channel does not have 3 waters. And if you try to make the channel conductive, but keep these water there, there's a clash. You can't. You can't. So these waters are pretty important to recover. And in fact, if you remove them, then the filter will recover. So that means that these waters are pretty important. So what we did is out of the information of the Anton run, which is like something on the order of 10 microseconds, it's the time for like 1 set of water to leave 1 subunit. That's about that. The time it takes for the waters to return in there is like 79 nanoseconds. We did that experiment like about 30 times. So these time scales come from the Anton. This also comes from Anton. If you have no potassium bound and you want the potassium to rebind to the filter, at 150 millimolar it takes about 8.3 nanoseconds. And if you assume that this is linear, like Szulkowski diffusion, then if you change the concentration, the time will just linearly scale with that. So if you go to 150 millimolar, it would be 8. If you go to twice the concentration, the time will be twice shorter. If you go lower in concentration, the time will get longer. If the concentration is 0, then the time is infinite. So this is just that. We made this little kinetic model that there's basically 3 parameters. And we simulate this, and the stars are the result of the simulation. And the red dot are experimental measurements for recovery. It's a function of potassium concentration. So you see that it matches surprisingly well. And this is 16 seconds. At low potassium concentration, this last step is more regular. It's 16 seconds. And the reason why it gets so slow is because you have to go all the way to this. But you know, the waters are bound there, and with these two rates, you can do that in your head. It's about 1% of the time you lose the water in one of the subunits, but that's 1%, that's 10 to the minus 2. If you have 4 subunits, it would be power 4 if they're all independent. So the probability to get there is actually pretty low. And so there's like kind of a statistical gating out of that that makes it very, very slow. And so that's why you don't, and we predicted that if you change the osmotic strength of the solution, you're going to, you know, we perturb the equilibrium of these water molecules. So we put big sucrose in the solution, and actually we accelerated the recovery because now we were sucking up the water. So we accelerate the recovery, even though the viscosity of the environment was 30 times higher, which is mind-boggling because normally it would be normal, it would be the other way around. So this is pretty nice. We felt really good about that. But we want to understand in activation too. So this is about the recovery thing. Once you're here, you close the gate, you recover. But the real thing that is in activation is you have a conductive pore that's open, and then you're going to inactivate. So it's going from this state to this state. This is a closed conductive. We have crystal structure. In water flows, we've got crystal structure. For example, we got several. But this is one example where the gate is open, but the filter is inactivated. So it looks like that. There's not really a crystal structure of this guy. We have to make a model of that. So to make a model of that, what we do is we take the filter of this guy and we sort of stitch it there. And then we equilibrate. So we did that. We started with a conductive open pore, and lo and behold, you know, in about less than 1 microsecond, this is the distance across the filter. So when the distance is large, you're conductive. When the distance is short, you become pinched. And you see, they all pinch. In about 1 microsecond, all these guys pinch. You say, okay, well, that's kind of good and not so good because it's supposed to take 1 second. And this is now 1,000,000 times too fast. Now, I know the force field is shit, but still, you know, 1,000,000 times too fast, that's kind of not good. You know, so, sorry for the French. No, no, but it's like, you know, we always blame the force field if something goes wrong, but this is not really, this is beyond the kind of thing that is just a bit wrong. This is deeply strange. So we were very puzzled. The thing is that at the end of these simulations, if you collect the densities of water, you get this mesh plot. The mesh plot is from the simulation at the end. The red dots are from the crystal structure of radiation and of the closed low potassium. I mean, we smash, and this is the gray, I think, is the crystal structure, and the color is the MD, or vice versa. It goes right to it. Right to it. It's unbelievable. You see, like, surely something is not that cranky about those simulations. What is going on? So we start to look at that and say, okay, there's a closed pore, there's the open pore. What if you have the pore a bit less open? Because Eduardo Peru got all these structures. He has many of them. So when the pore is partially open, according to its structure, the filter is conductive. So we simulated all sorts of channels. So this is the closed channel. This is open by 14 angstrom, 16 angstrom, and the 23 you just saw. So at 23, it pinches. At this is 16, it pinches a bit and then it recovers, you see? At 14, it does not pinch, and the closed channel does not pinch. So clearly, the opening of the bottom gate has a huge impact. I mean, it's not something we had sort of anticipated. We did a PMF. That's like NAMD, multiple copies, all that. So if you have the 23 inch, because that's kind of healthy open gate, and you do Fourier G map, this is the coordinates for the pinching, and this is the coordinate for the last ion that binds. Of course, the binding of ion, as in the recovery, the binding of ion affects entry into inactivation. That's known by physiologists for 30 years, 40 years. So that's why we control this with umbrella sampling, because we don't want to have like a loose averaging of the ion in the filter. So we do umbrella sampling on that too. And you can see when you have a gate at 23, the most probable part is here when you're at 5.5 actually. That's a pinch filter. But when you're open only at 14, you have 2 wells. And you could be stably conductive. So if you're just partially open, you could be conductive. And so This is the cartoon I sort of showed you before. That you could be closed, you open, conduct, you inactivate, I close the gate, I recover. I changed my mind. I think this was wrong. This is figure 1A of our Nature paper of 2013, but actually this is not right. We think this is what's happening. When you give the stimulus, the gate partially opens. And when we look at the dynamical motion of the gate, it's actually pretty slow. We think that you could stay around, and there's a lot of partially open states because they could be a bit asymmetric. You have force of units, so some of them fluctuate. So we think you hover there for a long time, and as long as you hover there, you're conductive. And then if the gate opens completely, you're gone. You're gone in 1 microsecond. That is a very different thing than what I thought before. I thought the open gate had a filter that was a metastable state in the conductive state. And that's not true. We think the gate has to be partially open. In fact, in KCSA, there's a big C terminal. And if you shut it off, the channel activates faster and deeper. Because the C terminal, it makes a bundle, so it keeps it together. If you shut this off, the channel opens faster and wider, inactivates faster. So it's very, very consistent. And so we think this is like a paradigm shift for us in terms of understanding this inactivation in these channels. And still, I have not a lot of time left. What time is it? 2:35. Am I supposed to start a little late? Yeah, okay. So I'm going to be like Klaus at his keynote address at Biophysicists. But not an hour and a half. Okay, good. Okay, so just quickly on the pump, it's very interesting. So we work on the pump, the calcium pumps. It pumps, these are P-type PPAs. It pumps 2 calcium one way. I thought it pumps 2 proton the other way, but people tell me they don't really know. It could be 3 protons. It could be 2. It's not, protons are hard to pin down a little bit. So this is a little difficult. It's very similar to the sodium-potassium pump that we have. But this is the one for which is all these structures from Paul Neeson and Toyoshima. And so we basically have all the states of the full cycle. It's just fantastic. And people call that E1, E2 pumps because there's a big family of all those states where you sort of more access to one side and the other family of states as you access the other. So it's the alternating access mechanism. And just to take the 4 dominant states, if you simulate them and you look at the water and this beautiful DMD rendering, and you remove the protein, you can see that this is accessible. This is not truly accessible. This is now accessible to this side, and now this is not accessible. So you get these this cycle. So we wanted to understand the cycle, like how you do the pathway between these. So you get crystal structures, but even though you get many of them, they're still pretty far from each other, isn't it? It's still pretty hard to do the pathway between them to understand the mechanism. So we proceeded in a kind of step-by-step procedure. We did, first let's do a coarse grain type of model, and then we're going to sort of take that coarse grain to fashion the atomic motion with all atoms. And so the coarse grain is based on something we did with Yvette Bahar. It's a kind of an isotropic model, but we have 2 states. And so you're able to find, this is like a C-alpha pathway of all the states. You're going to see some flash at the top here. So this is what the pump is really doing between these crystallographic states. It's moving around quite a bit. So these are not morphing. This is not like a visual morphing linear interpolation. These are the genuine pathway at the coarse grain level. So then we lift the coarse grain pathway at the C-alpha, and we use that in the targeted molecular dynamics to drag the all-atom. So we thread the all-atom systems along this nonlinear pathway of the C-alpha, and that gives us that. So this is now the pathway with all-atom. I'm not showing all the water in the distance, but this is all the pathway, and then there's a lot of, maybe I won't show the whole movie. This is the full cycle, though. Maybe I show you, like, what's going to open you. This is kind of dramatic. It's going to open you. See the water coming in. Web. Yeah, there it is. You see the water? It's hard to see what's going on with these pathways. It makes the paper extremely hard to write because there's like so much going on, you don't know what to say. So the conclusion really is that you bind ATP to this nucleotide binding domain, the green guy, and his job is to bring it close. So this is the ions. I'm going to come back to that. These are the calcium ions, and as this motion happens like this, the hydrophobic residue is like pushing out and occluding this, you see? So what happens is that the ATP is bound to this N domain, and they sort of come together to bring it close to S361, which is a highly conserved residue. And once this complex is formed, the calcium sort of at the same time binds to the thing, and the red domain here, you see, pulls with the linker. It pulls on this helix that will basically occlude the pores. It's kind of all concerted. So you go to that concerted occluded state. What's kind of more interesting is now the ATP is close to this S351. We don't do the chemistry there, but you assume that phosphorylation would happen. Well, what happens when phosphorylation happens is that now the domains, the red domain pushes the green domain and wants to interact with the phosphorylated residue. And by virtue of that, this linker is now pulling, like, so these are the occluded state, and you can see this is starting, and this is the bottom gate. Now, by pulling on this, the gate opens, and now the water comes in. Okay? Which is fantastic. So, again, this is hard to see just with the movie. If you look at the pathway, so we have, the string pathway is like a sequence of states. So it's a bit like a mini molecular dynamics. In fact, with the swarm, we have like 300 copies of the pathways, so we have lots of pathways. We can do everything we do is usually an average over many pathways because we have many of them. We start here, it's occluded, and it has ATP bound, and then it gets phosphorylated. If you look at the motion, what happens is that the red domain, once it's phosphorylated, will want to go bind to the blue domain. So these coordinates, this is the distance to the D51 and this thing in the red domain, will start to decrease. And then the distance across this gate will start to increase. This reminds you a bit of what we had in the kinase before. So this is the genuine pathway. In fact, the red dots is the pathway that came from the coarse grain system, and the green is after many hundreds of cycles of refinements of string metal with all atoms. And so clearly, this is what it wants to do, and there's clearly an intermediate at the bottom where the cytoplasmic domains have rearranged, like they move from here to here, they move here to here, they don't really move anymore. Now it's the transmembrane domain that moves. And so essentially, this reaction, to go from here to here, has to have this intermediate. There's a crystal structure of this guy. There's a crystal structure of this guy. I just came back from the Kyoto conference at Hiroshima. It's mostly in the honor of Hiroshima, but it's a big pump EPA conference. And Professor Suzuki was presenting data showing that this intermediate must exist, even though this is the only state that has not yet been crystallized on that cycle. Just before my talk, he was showing data that was showing that there is an intermediate. So I said, well, I have the honesty of my ignorance. I had no idea you actually were doing that. But clearly there is an intermediate. And then Paul Neeson was already thinking of tricks to try to crystallize this intermediate by playing tricks and things like that. So this is, I think, a winner for the string method from this system. One thing I want to just emphasize also is that the transmembrane part sometimes is the slave of the cytoplasmic domain. If you look at what they do, at the beginning you bring the ATP. Once it's phosphorylated, the red guy will shove this and will bind to the top. This is here, you see? And so essentially what you have is that you have three proteins. And this is looking at the bump from above, and always looking at that. And so the blue part is the domain that gets phosphorylated. It's like the surface of a protein. I'm going to be done. The surface of the blue protein is mutated or is modified by that phosphorylation. So without phosphorylation, it's a green domain that wants to bind there. That's just protein specificity. It likes to do that. Once the phosphorylation has occurred, the best binder is not the green domain anymore. It's actually the red one. So it pushes it and it binds on top. So it's essentially just a change of the protein surface specificity that activates that. And once the red domain has done that, it's then that you drag the transmembrane part. So, so we started to look at the free energy of the movement of these domains with and without phosphorylation, with umbrella sampling. So this is with ATP, and when you have ATP, this is the state that should be the most stable. But when you have aspartic 351 phosphorylated, this is the state that should be the most stable. This is only 12 nanoseconds per week. This is the only thing we could burn on blue waters. But we just got an insight of, I don't know, 95 million SUs or something like that. So we're going to basically burn that. I'm pretty sure this PMF will improve, and that this state will start to become much, much more stable. And you can see that, you know, this will inform us basically on the thermodynamic of moving the cytoplasmic domain. And this is really what powers the transmembrane part. So this is really what I think that should be done. I'm going to I have like just one thing about the proton stuff that I'm going to skip that. So, thank you. I'm going to answer your questions if you have any. Thank you.
Any questions?
Yes, so regarding your PCSA talk, how general for the mechanism for inactivation you are for any other channels? I understand that the inactivation mechanism for, for example, calcium channel, sodium channel is completely different as like
True. Well, inactivation happens in all cytoplasmic systems, so enzymes, whatever. But I think for potassium channels, so I think it's important to basically separate potassium channels from others, because sodium channel activation could be quite different. Their filter is different. Potassium channel inactivation was first observed on voltage-gated potassium channels. That's the shaker type, etc., etc. This has been a phenomenon that physiologists and like Chris Miller, you know, and Clay Armstrong and these people have studied since the early 70s. And so it took a kind of a mystical view. When the first crystal structures of KCSA appeared, it was mostly about permeation and selectivity. After that, there were more crystal structures. Some of them have an open channel. In fact, the only channel that looks kind of inactivated is the KCSA. The first one was at low potassium, and now in Barroso 2010, got many structures with the open gate where you see it's constricted. So this has led people to think that maybe KCSA is an outlier, and the mechanism of inactivation in the real KV, the voltage gating channel, must be something different. I would counter that with two arguments. You know, I would say, first of all, the pore domain of KCSA is 38% identical to shape. 38% identical. This is like in my cousin meeting. Identical. So, this is not a little bit. You know, like in most other fields of proteins, for example, in kinases, people talk about Src kinase and HCK, and then they observe something enable, like the DFG flip, and then you come back to Src and say, oh, maybe the DFG flip. I mean, you talk to people who are like John Currian or the people in the industry who do kinase inhibitor, people think of the kinase as a family, and if a motion really or a confirmation exists in one state, in one of the member, they wouldn't necessarily rule out. But the ion channel field is a little bit, you know, some like semi-religious, but every channel is really a person, and you cannot imagine that KCSA could really, this bacterial channel, could inactivate the same way as shaker, even though that seems to be pretty possible. So why is it that no crystal structure of these KV do not look like they're pitched? Because there's the KVAP that was crystallized, it's open and it's not pitched. There's the KV 1.2, so KV 1.2 electrophysiologically does not really inactivate them. So it makes sense that when you crystallize it and it's open, it's not really inactivated. KVAP is not inactivated, but it's kind of busted because its voltage sensor is falling apart. So we think actually it's kind of a relaxed open state. But if the voltage sensor was really trying to open it, the gate would be open a bit more. And according to our calculations, then the filter would pitch. And the place where the transmission is done is the residue just between the filter and the body gate along the inner helix. If you have a large residue there, in KCSA has a phenylalanine and an isoleucine, two large residue there, the coupling is stronger. If you make alanine there, the coupling is gone. And in case of shaker and KV1, these are pretty small residues. So we think if we put larger residue there, you're going to probably promote inactivation. So I think actually the KCSA is an absolutely acceptable model for inactivation for KVs. But it is still a bit controversial. There are people who find that not yet compelling. And so, you know, the lesson I have is that when there is that, you have to have more data. It is not just like a matter of screaming at each other. You'll have to have more information to convince people that you're right. This is not just a debate where you bang your fist on the table. I mean, we'll have to have more information.
All right, let's thank the speaker again. And our next speaker is Sharon Hammes-Schiffer from the University of Illinois.
This morning we are speaking with Klaus Schulten, PhD. He is delivering the 2015 National Lecture. He is with the NIH Center for Macro Molecular Modeling and Bioinformatics. And good morning and congratulations. This is the highest honor offered by the Biophysical Society. So congratulations to delivering the speech today.
Thank you very much, Marianne.
Give us an idea a little bit about what your topic is going to be.
So as the title of my lecture states, I want to show that computing today, just like experiment, leads to discoveries. So we, in a way, in what we call computational biophysics, becoming competitors to the experimental colleague in the sense that we also want to make discoveries and that we have made discoveries. And that is what I want to explain and showcase in my lecture. So we are actually both partners, but also competitors to experiment. We have the advantage that we can see processes where experiments very often can only see static structures or static averages. And so we work with experiment to complement the experimental data by explaining what they mean in terms of the underlying biological processes, very often today the same publication. But we often also play the role of integrating data, sometimes together with computational models. And there we play a role that without the computing there would be no discovery, because you have to first integrate the data to get the result, and that is where we play an important role.
So that's fascinating. It's so widespread into so many facets, but then taking it into a practical application. How can these other scientists today take that information and apply it to their specialties?
So the first have to make discoveries to know what is happening, and in biology and in biomedicine we have to realize that basically all organisms are large societies of molecules, and every disease of a healthy organism relies on the molecular processes, and this is where the computing plays a role at a kind of microscope. We always describe processes that occur at the molecular level in organisms, but in the past it was just virtual reality, and now it is becoming so accurate that we really dare to call ourselves a microscope. And this microscope shows events that are, for example, very critical in medicine. Alzheimer is an example where molecular events that cause the disease, many brain pathologies that are not understood at all yet are clearly related to molecular events. You go usually to the pharmacist and buy some tablet. That's the most common medical intervention, and that also tells you that these molecules, stupid, that really make the body, and we need to explain it, and we need also we need to know about it to assist medical treatment.
There has been debate, or you might say challenge, as to how biophysics is actually defined. What is your definition?
So I'm a physicist, so I sometimes use the word biological physics as opposed to biophysics, emphasizing that the physical methodology and the concepts are most important for me when I approach the field of biophysics. So that means that in order to understand organisms from the electronic level to the atomic level, and then all the way to the cell and to the organism, I utilize the latest and the best physical concepts, but also the best physical methodologies that permit us, for example, today, to look into the living cells and to resolve the chemical detail in living cells. I'm a co-director of the National Science Foundation Physics Frontier Center with the title Physics of Living Cells, where we basically bring the physics to the cell in order to resolve processes that occur there, and again that have very clearly biomedical implications.
And thus the merging of biophysics, bringing them together.
That's right.
All right, Professor Schulten, thank you so much, and we look forward to hearing your speech today.
Thank you.
All right, we're ready to start because I'm such a nice guy. We're 25 minutes behind schedule, and I assume that we wine at the reception. So I'll ask our speakers to show some self-control today. And you have officially, 40 minutes, including questions. And our first speaker is Neil Hunter of the University of Sheffield.
Okay, well, first of all, I need to say how privileged I feel to be giving a lecture at the Symposium. So, I'm going to talk about that aspect of Klaus's work which concerned photosynthesis and energy and electron transfers. It's remarkable that he was so versatile in his career that so many of us here can give completely non-overlapping talks, and Klaus made a fundamental contribution to all of us. So there really is very little overlap between my talk and anybody else's, but still, despite that, this particular work did occupy a soft spot for this work, I like to think. So the inspiration for the work, by the way, that I do, that Klaus supported, was the input of energy into the biosphere, 100,000 terawatts raining down on the planet, a small percentage of which, through the agency of chlorophyll, is filtered into the biosphere. Around 10 to the 33 chlorophyll molecules on Earth, so part of my lab actually works on biosynthesis of chlorophyll. And these numbers are pretty compelling. You only need 1 hour of sunlight in principle to fuel all of mankind's needs. And every year photosynthesis converts around 200 billion tons of carbon dioxide into useful things like food. Alas, they can't quite keep up with the amount of carbon dioxide that's being produced, so it's starting to build up in the atmosphere. And at the same time, the excretion product of photosynthesis, oxygen, is immensely useful to us as respirers. So it's very, very important, photosynthesis. And just a very brief summary of what we do in my lab. We look at the biosynthesis, we look at the enzymes of chlorophyll and carotenoid biosynthesis, we do enzyme kinetics, pathway engineering. We look at the assembly of these proteins and how the newly synthesized chlorophylls are delivered to the assembly machinery. We look at the structures and functions of the membranes and complexes involved. And finally, finally, we do a lot of bionanotechnology where we imprint these molecules on surfaces and try to create sort of new energy transfer and electron transfer pathways. And what we're trying to do is to understand photosynthesis in terms of the architectures that make light absorption and energy transfer possible. We want to understand how the pigments are synthesized and assembled into the complexes. And then with Klaus and Nelly's help, we want to construct all atom 3D models of photosynthesis. We've done it for purple bacteria, cyanobacteria and plants. Well, we better get a move on. We've got about 4 years left, so Nelly needs to hurry up. But we are ideally poised to do this to cyanobacteria and plants as well. And then we want to find out design motifs that emerge from these computational studies and try to construct new nanofabricated materials that will do this kind of work. And the synthetic biology side of the work is to design new bacteria to harvest sunlight. So the bacteria that we'll start with are the purple bacteria. I'll get on to cyanobacteria and plants later on. The purple bacteria can be thought of as the sort of the E. coli of photosynthesis. They're gram-negative bacteria that elaborate an internal series of vesicle structures which house the photosynthetic apparatus. And these have been the sole focus of our work with Klaus. These bacteria can be found in stratified depths in freshwater lakes. They can be grown in the laboratory. In big bottles, you don't need salt and all that messy fancy type stuff. So when you slice these things open and do tomography, you can see these particular structures. And these small molecules, small structures, around 50 nanometers in diameter, and colloquially called chromatophores. These chromatophore vesicles, we'll see a movie of these in a minute. They can be thought of as little independent bacterial organelles, since they discharge the central function of the cell, which is to convert light into ATP. Each one of these is an independent little unit, like a little light-driven nano-machine. So the basis for the work with Klaus started in 2005. Nelly Senna, who's the instigator of all this, came up to me after I'd just had one of my very, very rare successes and had a paper published in Nature on the atomic force microscopy of these bacterial membranes. And I must have given a talk on it, I guess, at this Gordon conference, and Nelly came up to me in the bar and wanted to, he just kept pummeling me, asking me questions. And we formed this collaboration, and Klaus absolutely loved it and put all his formidable intellect and, let's say, funding behind it as well. And my contribution as a biochemist was we did the quantitative mass spectrometry to count every protein, every relevant protein in the membrane. We've done a lot of electron microscopy and X-ray crystallography to determine the structure of this membrane protein that you'll see in a minute. Then we've done the atomic force microscopy to see the locations of these dimeric photosystem complexes in the membrane and also these LH2 complexes here, so we could start to sort of piece things together. Then lots of other people in lots of other laboratories have done a lot of kinetics to look at the exciton dynamics within the system, charge separation within the reaction center, electron transfers in the BC1 complex. This was coming across a lot of this. And essentially, this whole system, kinetically, was quite well described. It was simply that people hadn't known the stoichiometries rigorously, nor had they any idea of where everything was. But finally, because we could place everything in a map, this is when Nelly was let loose, and then he started making these 3D membrane models. And I put this slide in because some people wonder why you should even care. Why, because these, by the way, these things here, you see these in textbooks, and there's always one of these, one of these, one of these, one of these. And, that's not going to work. So why do you need to know stoichiometry, but in particular, why do you need to know where stuff is? To me, it seems obvious, but the fact is that you need an extensive network of some of these molecules, the light harvesters, because they're acting like a satellite dish to harvest the solar energy. You need a high quantum unit of charge separation, but this requires an optimal number and distribution of reaction center traps within this light harvesting matrix. And of course, the number and distribution of cytochrome, or cytochrome BC1 in this case, complexes, ensures that the migration of diffusible components in the membrane, namely quinones, isn't hindered by crowding in the membrane. So, you know, it's not a completely accidental random distribution of protein, as we will see. So, Melly's first model was published in 2007 in PNAS, and but, you know, I don't want to give a long talk if we can possibly help it, especially since we've got time. So, this was the sort of the most recent useful iteration that was, you know, this is subsequently there's a paper in eLife which is much more mathematical. But the fact is that a lot of the basic aspects of that model are in this earlier paper, and we can see that this is the single 1.9 million atomic level model of the chromatophore membrane with the cytochrome BC1 complexes here in purple, the dimeric reaction center LH1 complexes, and the matrix of LH2 complexes, and these are the workhorses for absorbing solar energy. And they're going to feed it to these complexes here, and then these are the ATP synthetases. And so there's this, Professor Baumeister used this word concerted, I think, but so this is like a concerted series of energy, electron, and proton transfer reactions, all linked together. And I should also say that missing from this generation of the model are all the lipid molecules and water molecules, but that takes it to 100 million atoms, and you know, that's crazy. So it's just that's over the top. So this is the modest 1.9 million atom model. It's still very useful for actually, it's able to account for every single process from photon to ATP, every process, every kinetic, every stoichiometry, every structure. Okay, Stu. So Klaus was really passionate about this thing because he loved this possibility to turn the whole thing into a movie. And actually, you can see it on YouTube. It's a very nice teaching tool, and the movie itself is actually quite a large file, so I tend not to play it in PowerPoint now. There's no point really. I'd rather play it in some kind of media player. Actually, not Windows media player. Anyway, but look, I'm going to play it separately out of PowerPoint. Hopefully, it'll play smoothly. And it starts in a charmingly rustic fashion, but don't worry, it gets more scientific. So basically, these are, this is a Biochemistry story cell. We're going to zoom in on a single chromatophore. There are around 300 or 400 of these per cell. And in low light, the cell makes more of them, and in high light, the cell makes fewer of them. And so here we are, this is this chromatophore. This one represents 2,577 aperture chlorophylls, and around 67 of these green LH2 complexes. The LH2 complex structures determined by Richard Cogdell and co-workers in Glasgow in 1995, and is a ring-like structure with 27 aperture chlorophylls and 9 crotinoids. An excitation of the crotinoids, for example, will lead to an excitation transfer to the chlorophylls. If the chlorophyll is absorbed by it, it can transfer it actually to the crominoids, but the fact is that after a few femtoseconds, the energy is equilibrated around the ring of chlorophylls here. So these act as little temporary storage rings, and the energy can migrate by first the hopping from one ring to the next, and then finally to the reaction center here where it's a charge separation. So This was Richard Cogdill's structure. This is my structure here. It's actually one of the largest membrane protein structures. 80 transmembrane helices. Published this a few years ago. And this is the ring of LH1 bacterial chlorophylls. They've taken the energy from the LH2 chlorophylls in the complex. And it's interesting, you've got this S-shaped ring of bacterial chlorophylls. The energy will migrate over a long distance, 25 millimeters, sorry, to the special pair here. So you get a charge separation of the special pair, and then migration of electron to quinol here. After 2 successive turnovers, you get 2 protons and 2 electrons give you quinol, and the thing leaves the reaction center. So everything so far has been about making a quinol. Everything, all the energy transfer, electron transfer, everything is about making a quinol. This quinol will migrate to the cytochrome BC1 complex. I'm not going to advise myself by getting it mechanistically wrong with Tony Cross listening. But what I will say is that it's a very clever cycle mechanism. You would think in theory that if you had 1 QH2, you'd only get 2 protons. You get 4 because the key cycle effectively amplifies the process. And you get a bifurcated electron migration within this complex. The bottom line is that you would get 4 protons and proton motive force generated in this cavity here within the vesicle. So, the cytochrome BC1 complex is situated quite near the reaction center, but not part of the super complex, absolutely not. The electrons from the BC1 complex will migrate to fill the electron hole within the reaction center, and the protons, they will go to turn the rotor of the ATP synthase, and this is sort of back to John Walker again. And effectively, the CAM inside the ATP synthase does mechanical work, and the bottom line is that what you're going to end up doing is turning your proton motive force into chemical energy in the form of ATP. So the whole thing is an assembly that does a concerted series of reactions from photon right through to ATP. And so it was represented nicely in the movie. Don't like the idea to do it, it was very nice, quite elegant, and so they represented all this as a sort of series of interlocking wheels turning. So you've got your sort of the light harvesting bit, turning the electron transfer, turning the proton transfer, and then the ATP synthesis. So this was a huge amount of work. I mean, I can't tell you how much work this is, just crazy. And all I was the sort of the experimental monkey who provided some of the data, and other people were also amazingly helpful. Tony definitely was, Tony Crofts, and other people as well. So that played at double speed, by the way, because it really, when it's at its normal play speed, it's what you might call stately. Too slow. All right, so get back to the tool. Oops. Okay, so these were some of the papers. As I said, the first one was in PNS in 2007. We all know that Klaus gets more citations in one single year than most people would include in their whole life. And this is the only paper, I suspect, that even has a fighting chance of contributing to Klaus's H-index. I think it's been cited about 150 times, and that probably just qualifies it. None of the rest do. But I'm proud of giving Klaus one score on his H-index. And then all these other people, of course. I would say it's an awesome piece of work. One of the interesting things about it is that it made some clear predictions that actually the limiting system, the limiting factor in the whole process, actually is the number of cytochrome BC1 complexes. you know, if you have a series of interlocking reactions like this, something's going to be missing. It turns out to be this here. And I was able to do a simulation to show that the number of cytochrome BC1 diamonds per vesicle is effectively scaling linearly with the amount of ATP production. The other interesting thing is that the way the system is configured, that actually at 3% of bright sunlight is working at 50% of its maximum rate. Quite nice. This means that this interesting vesicle system is actually evolved specifically to function in low light. Okay, so that kind of, it doesn't finish the work with Klaus because actually we're still working together. It's quite a nice thought really, where there's still papers to come out. But this is really was Melly's brainchild. And so Andy in my lab did the experimental work, and there was this slightly outrageous suggestion that just because you knew about this one vesicle, somehow you could predict cell doubling time. And it seems crazy to even take something like this on. That's why you need to work with crazy people, so Melly did it. And basically, starting from this atomic detail structural model, you can compute the ATP production rate per vesicle. And then there's this concept of return and investment time. In other words, how long does the chromatophore take to pay back its own student loan, or whatever you want to call it. And then the cell doubling time can be expressed as the time it takes for the whole cell to pay the investment necessary to construct a new cell. An interesting thing was that the return on investment time is, for a typical illumination range here, corresponded to cell doubling times of that. So the return on investment times and experimentally determined cell doubling times actually, they weren't too far out. It's actually a remarkable piece of work. Anyway, we must move on. So now cyanobacteria. So cyanobacteria, they really are important globally. The Proton-bacteria less so, but cyanobacteria may do as much as half of all the world's photosynthesis, and of course the reason is the planet's 80% water. And so these are some of the workhorse strains Synthesis here, and you can see in this animation the cyanobacteria in the oceans sort of pulsating year by year. Interestingly, you can see some parts of the ocean, like this part of the Pacific, is almost barren in terms of photosynthetic productivity, famously because of the lack of iron. This Photoreceptor was only discovered in 1996-ish by Sally Chisholm at MIT, and this is the most abundant species of anything on the planet. And we can also see another cyanobacterium here. This isn't cryo-tomography. This is work done much earlier with vector staining. But the point is, you can see that these bacteria don't make vesicles, they're making these layered sheets of membrane. And in particular, we wanted to learn more about photosystem 1, and that's because photosystem 1 provides reduced ferredoxin for the cell, and that's really important for lots of nitrogen metabolism in the cell, sulfur metabolism, hydrogen metabolism is absolutely essential for all of these processes. So we took, actually not this cell, but we worked on a bacterium called Thermosymphococcus elongatus, and we took the membranes out of the cell and we broke them into flat sheets. And we used atomic force microscopy, which I was almost embarrassed to show this when Professor Gow gave a pretty good lecture. Anyway, maybe I will show it. Well, look, the thing is, this isn't single molecule force spectroscopy, this is mapping, so it's slightly different. So basically, all it is the AFM tip is raster scanning across the rough surface of a membrane, feeling the bumps effectively. And as it does so, it builds up a topographic image. It's actually a 3D image because actually height is the most useful thing you get from this. And this is done ambient temperature conditions, ambient pH, and high signal-to-noise ratio. You can actually fit the positions of some of the complexes in here, but you know, it's a bit of guessing. So what we do is, in this case, at least we can say something about the heights, because that's AFM strength actually is in the Z measurement, and so we could tell the difference between cyclone B6F and photosystem 2. Then we had a different biophysical method as well, and we used an adhesion-based method to map the B6F. But back to PS1 anyway. So we found, to our surprise, that there were these patches of membrane with these exquisite trimeric arrays of trimeric photosystem 1 complexes, and we're pretty sure there were. The atomic structure of PS1, and everything completely matched. We were also able to measure the heights, and because we know the atomic structure, you can place all the cofactors in relation to the membrane bilayer for the very first time, and you don't get that kind of information from X-ray crystallography. And then Melly was able to read his magic, and then what we could do is interpolate the PS1 trimeric structures into the membrane map. So this is the topography map here, and you can see it gradually giving way to the complexes, and you could even orient them quite precisely. And then Melly was able to compute the path that energy must take within this array and show, in fact, that there's a reasonable possibility of energy transferring between photosystem 1 complexes, although the great majority of the energy that hits a photosystem 1 will stay in-house, because photosystem 1 is very fast to track, and most of the time the energy will just stay within this trimeric track. But there's a finite chance that within that array, energy can migrate between trimerals. And in the same paper, we did super resolution this was structured illumination microscopy. We labeled photosystem 1 with YFP, and these images clearly show that you can see the localization of patches of photosystem 1 in linear cells. So this was published this year with Klaus on it, and yeah. So I'm going to finish with just 3 things. I'm just going to finish with 3 things that I would have liked to tell Klaus about. So I would have liked to tell him about this. So it became possible, in collaboration with Jamie Hobbs at Sheffield, to image these in a new way. Previously we had to flatten them out and squish them onto a flat surface to do the AFM, which isn't great, but see what you can do. But with the new AFM that we had at Sheffield, it was a very, very, very patient guy, so it was possible to image intact chromatophores. So these are fluid-filled balls, but this pointy thing is doing this, and we just move around or squish. I mean, it's a horrible thing. But with the new AFM, we could image intact chromatophores, so this was the problem. And these are the images, but when the high-pass filtered, you could, for the very first time, see the LH2 molecules in the intact chromatophore ball, and you could actually start to see core complexes with this single reaction center there. And then we analysed 180 chromatophores, and we were able to get statistical distributions of all of the spacings between all types of protein within these intact structures. And we were even able, on occasion, it's true, but we could actually image the single ATP synthase, but this is very hard. But they are there. You can see the odd one within the chromatophore membrane. There's generally only 1 or 2 chromatophore anyway, but we could see them. So that's, I think that would have been nice, because I think Klaus would have liked that, because, it agrees with the model anyway, which he would like, and it would have been the basis for a refinement of the model. What else would I like to tell them about? This paper has just come out in Nature Communications with Greg Engel. And for the very first time, it's been possible to use two-dimensional femtosecond spectroscopy to look at small groups of living cells and actually measure. This is the first time it's been done. I'm not claiming credit for it. It was Greg Engel, Pete Dahlberg. Again, I'm just the grunt that gives them that material. But this is really amazing. I mean, technically, this is an absolute tour de force. To actually be able to see all of the things that have been modelled by Nelly, but then be able to reconcile it with the whole cell and the energetics of the whole cell, I think Klaus would love this. So actually the data that Greg Engel and Pete Dahlberg produced, like I said, the first measurement in the living cell, and they were able to deduce from first principles a model which, qualitatively at least, is very much in agreement with the atomic level model in Klaus as well. And actually now it's capable of extension to more complicated photosynthetic systems. So I think you would have liked that. And then finally, we've started, we've been working on plants for a long time. Again, it's this membrane organization problem. Efficient energy transfers require proximity of chlorophyll protein complexes, and electron transfers require fast diffusion of electron carriers. So, how are you going to solve that problem? And so, again, it was an atomic, for us, this is an atomic force microscopy problem. So we started working on spinach. That's an Arabidopsis, but anyway, it's a plant. And so we started working on plants. Spinach we work on, I'll be honest, the reason is because you just get it from the supermarket. You'd have to grow it. You just go down to Tesco or, you've got more money, you go to Waitrose, but whatever it is, you just get a bag of it. A few hours later, you've got your stuff. It's laughably easy. And this shows the interest of the system, because actually 400 square centimetres of membrane are found within a square centimetre of leaf. So there's a massive amplification of light absorbing capacity. This is just from a textbook, but basically you get these chloroplasts and you have these stacks of pancake-like membranes interconnected by these stromal lamellae. You can see them here in this negative stain image. And people have done tomography on them, very cool ones, and more spectacularly Benoit and Baumeister. And so we wanted to see what these things would look like by and this is your idealized electron transfer chain. It's one of these, one of these, one of these, and one of these, and they're always there. They're not. But this is where it's always depicted. And the idea is that photosystem 2 is taking solar energy in, it's giving you that waste product oxygen. Proton gradient is consumed by the ATP synthase. This complex is the analogous one to cytochrome BC1, and it's generating the proton-motive force. Plasticine is taking the electron over to PS1. In purple bacteria, this will go back there and fill the electron hole, but it's a linear system in plants, and photosystem 1 is generating reduced pyridoxine, and then you're able to reduce NADPH and fix carbon dioxide. So how are they really organised? And we focus on these two complexes here, and these are some AFM topographs of Brana discs. These are 500 nanometers across. And let's say you see lots of dimeric particles. These are photosystem 2, and some of them are TEF complexes. And we used a method where we attached plasticine to the AFM probe. And if the plasticine is oxidized, and only if it's oxidized, and if the cytochrome BC is reduced, and only if it's reduced, you actually can see adhesion between the two proteins. You actually get force curves. And if you use this particular AFM in peak force quantitative and mechanical mapping mode, you can generate a false map of the whole membrane and find out what the cyclohexane complexes are. And so that's what we did. And basically, you can quite cleanly assign the different complexes to the NFM topographs here. It's published a few years ago in Szabo. And then the challenge for people like Melly is to construct a map of the whole ground membrane. One of the things that did emerge is that the cyclic V6F complexes are always quite near photosystem 2. Some people didn't think this, but actually the situation is almost exactly the same as it is with going back to steroids, and it's this concept of quinone confinement around these two complexes. This has been shown kinetically by Wolfgang, but then in this case massively overinterpreted to be a supercomplex, but it's not a supercomplex. There's no question that this system requires close proximity between V6F and PS2 components. Then these remaining components are found in the stromal lamellae. We've done a lot of work to model the membranes. We finally figured out how to make nice, clean stromal lamellae membranes. We've done all the AFM. We can see where everything is. This is just gone back to Nature Plant. And so now we have a map of the stromal lamellae and the interface between the stromal lamellae and the dry membrane, mapped by AFM. And we've done all the simulations for plasticine and plastic diffusion as well. So a complete kinetic model now for photosynthesis. And I'll just finish these. So these are my papers with Klaus. So this was the first one, I have to say. This is the result of the meeting in the bar. There were several things on various things, including membrane curvature and things like that, some jump down. And this final one. But there probably will be more. I really hope so. There's no question that, Klaus has a lot more mileage in all of this. And just to thank people in Roux, my sponsors, and of course, above all, these two people here, Melly and Klaus. Thank you.
So since we're 20 minutes behind schedule, maybe one quick question?
You were 20 minutes before I started.
It's not your fault.
So now that you've really determined all the components and the sort of geometry, what's the general rule of how often you have to have the ATP synthase with respect to the other components of the network?
What you mean in terms of stoichiometry, how you actually need?
Right. Instead of the picture 1.1, I don't remember the back and forth when the first looking at this, we only had one ATP.
Yeah.
Suddenly I looked at the picture, a year later, I go, where was it going to come from?
1, 2. What the number in the end turned out to be 2 from my spectrometry. But before that, some kineticists, including Wolfgang, had predicted 1. But it turned out to be 2. Of course, some of the time will be 3, some of the time will be 1. It turns out you only need 1 or 2 to consume all the protons you could ever make in the area light intensity. The potential turnover number of ATP synthase is around probably 600 or 700 per second. And I mean, that's good enough to sort of hoover up all the protons from all of the rest of the machinery. You don't need 10. Okay.
All right, great. Thank you. Next speaker is Ron Albert of the University of Texas in Austin.
Thank you. It's again an honor to be here. Before I start my talk, I need to tell some short Klaus story. So about 20 years ago, I We had a nice workshop at the Institute of Advanced Studies at the University. There are many good people there. Klaus, of course, was there. And the format of this workshop was rather unusual. It's an open-ended talk. You talk until you don't feel like it anymore. The only rule of the game was that you have to answer in full any question from the audience. There's no such thing as explaining you later, maybe we'll do it over breakfast or something. You really have to answer every stupid talk that somebody asks you, and both the person who asks the questions and the speaker should be completely satisfied. And it went quite well. I mean, this was a relatively small workshop, and there are talks of 1 hour and a half, and then people were just tired and went off. Martin Karpus gave about 145 minutes talk, in which at the end he sat on a chair and answered the questions because they couldn't really stand anymore. The only person that we really had to stop because we had to go to lunch at some point was Klaus. So that's the passion of Klaus. I sort of learned at that particular time, and I've seen this many other times as well. And I will show a connection to various points of the things that people are interested in and how we influence significantly our work by thinking on what we are doing for many years on thinking of reaction path and kinetics and the first passage time, which are tools in which you are using to study kinetics in biological systems. Since, they are 20 minutes late, and I'm not sure I make it to the end, let me put the thanks at the beginning, so the people that really did the work, there's Alfredo Cardenas, Juan Embelo, that now is in Princeton as a postdoc, we already did work on Antax that I'm going to describe, and Mangesh and Susan are our collaborator from Sandia. So, something that I believe in is if you want to study biology, you really must study kinetics. Everything that happens on the cell is really determined by rate, and the cell manipulates speed of different reactions by enzyme and whatsoever in order to get the process that it wants. And if you also think about the ever-growing field of biology, it's really about reactions and how you manipulate them in order to get the response that you want on a cellular level. In fact, system in equilibrium is dead. Okay, so we really must think about kinetics, and if we are studying biophysics and biochemistry, we are facing the challenge, and that's something that we are still working on. And it's not solved, thank God, because then I have nothing to do. But it's a challenge that how you extend the time scales to look on at the molecular level, at the atomic detail, at extremely slow processes that can go up to an hour. So if we are looking today, we can sort of do quite efficiently microseconds, but suddenly we are very much interested, like in the cell processes or in membrane permeation and other type of reaction in processes that are going to extremely long time scale. If you need now a single number that sort of captures a lot of the kinetics that happen in the system, what number you are going to give? So one possibility is to say, okay, I'll give you a rate constant. So I say no. It should be the mean first passage time that is defined as the average time in which it takes trajectory that starts in some place, for example, at the reactant, to make it to the product. So we have many trajectories going for the reactant to the product. We calculate the average time of this trajectory and we report the so-called mean first passage time. The advantage of these characterizations of kinetics that it captures also diffusion processes or non-exponential processes indicating a single number, which you cannot do really with the rate coefficient that usually corresponds to exponential time decay. So we are going to be after computing the mean first passage time when we discuss kinetics. This idea is definitely not new, and I can go that our Sharon here and Daniel here, as well as Klaus, actually published a very, very beautiful paper in which they are looking at diffusion reaction using the Minkowski equation and looking in very different aspects on the first passage time that help us think and formulate the process in a correct way. So this is at least one of the earliest contributions that I learned from on the mean first passage time by looking at the paper by Attila, by Sam, and by Klaus. Taking this into the world of simulation is of course a challenge, but this thinking about the MFPT, this is a recent publication by Klaus and his students when that
that essentially use very similar approaches to the kind of, in many cases even the same approaches, to the kind of approaches that I would like to advocate here, and looking at a very complex biological molecule. This is 85, all the everybody before, and they essentially looked and characterized the energy landscape and the time scale in a full atomic detail. This can be one of the significant achievements that made this possible, something that we have many times already today. Is indeed now. This is a beautiful software, and some of you may know that I, for many, many years, I developed my own kind of my own lab code. But recently, in about last year, I decided to give up. I cannot compete with NAND anymore, and I just been converted. So we are using NAND. Actually, the calculations I'm going to present were done on NAND. The other kind of tool that we are going to use is just using still molecular dynamics in order to get at least a rough description of the reaction coordinate in order to compute things after that. And this is another tool that was used also in the paper with Klaus and Ma 85, and also on the system that I would like to put forward now that we studied quite recently. This is another Ma. This is my Ma, his name is Pierre. And we study the anthrax channel. It's a pretty nasty system, but it's also a fascinating machine. It includes essentially 7 monomers that aggregate here together and form this kind of a tube of a channel that you see here. This end of the tube then punch a hole in the membrane, and what it does, it transports protein that the name lethal factor suggests what it is going to do. The protein is unfold, it passes through the channel, take it into the cell, and then do a lot of kind of a nasty things inside the cell. The first step in which the protein starts moving inside the cell is there is an unstructured N-terminal that you see in yellow and red in here, and that presumably permeates into the channel in here. It is not seen in crystallography, so it is really this older piece of the protein. But if you cut it off by such directions mutagenesis, the protein is unable to pass through the channel. So it is essential for the entrance of the protein through the channel and making it into the cell. The question is really what makes it possible for it to permeate and form a hook in the channel. After that, it is enough time for the protein to unfold and follow this N-terminal in here. So we were interested in looking at what essentially drives the unstructured N-terminal into the channel and leave it at the end there hook, such that the rest of the protein will be plugged in. This process can be very slow. It can take milliseconds in order for this just N-terminal to get in, depending on the pH condition. And we're looking here on the system, in which here is the low pH, and here there is a neutral pH. So, we're interested in the questions of time scale, and we had sort of a very simple idea. Here we have a reactant, here we have a product, and we have some way of some reaction coordinate that sort of capture the, if you like, the N-terminal out, the N-terminal in. And what you will do, if you like to use NAND, and to run a trajectory that is going like this, and running it back and forward many times, until it's finally into the project time. Even with NAND, we cannot do it in a straightforward fashion today. Calculating millisecond trajectory and many of them in order to get it kinetic makes it very, very difficult. So the simple idea is, if instead of running very long trajectory, we should write short, right? I mean, with a short trajectory, I can run in parallel, I can run the really very short trajectory, and somehow I need to combine them in such a way that we give you the correct rate. That's the trick, because you don't really know how to initiate trajectory that starts from the middle of the reaction coordinate in here, and how to collect some data from it, such that it will give you the overall rate and the overall kinetics correctly. So, that's essentially what we are doing with our theory or our procedure algorithm. It's called milestone. We're running a large number of short trajectory, and short trajectory, I really mean short. It's our picosecond trajectory versus predicting at the end of the day, sometimes in an hour time. And in the systems of anthrax, we are really looking at a system that is also quite large, about 700K. We're dealing in NAND in this case, and we are using still molecular dynamics in order to determine the initial reaction coordinate, and then using this short trajectory in a way that I'm going to describe. So,
I'm truly impressed by all this big system that we have seen today, but in order at least to explain and to appreciate what is involved in milestone, I'm going to do now first a toy model in which you can sort of look and understand everything what it is going. And this is a two-dimensional system that we have in here. It's a double well system. We have a well here, we have a stable state here, we have a stable state here. And here we have a narrow channel. The channel is actually only an entropic barrier, it's just a narrow pipe, such that if you have a trajectory that is running on the left side of the well, the left well, if you have hard time to hit at the correct time, the channel will pass to the other side. So this is very bad because the system is properly trapped at this position until it succeeded to pass for this particular hole. The question is, can you use this idea of using very short trajectory and the reaction coordinate in this particular case in order to speed up the simulation and understanding and kinetic and thermodynamics? So choosing the reaction coordinate is a critical step in many physical and biochemical processes. At the moment, for this particular system, I have it, right? I mean, I really want to go from here to here. I'm going to use the x coordinates. The questions of the reaction coordinates at the moment, I do not consider. When you work with using reaction coordinates, some of them we have done, some of them Eric van den Enden has done. There are NEB, there is string, there is what we call locally updated Planck. So, but it is still a challenge, and it's not a trivial issue. But at the moment, again, I said that we have a reaction coordinate. The milestone, or the way that we divide the space to initiate the short trajectory, is done by putting the hyperplane, or in this case it is a line, which are perpendicular to the reaction coordinate, and essentially dividing the space into effective cell. What do we do now? This is a movie that prepared by Juan Benoit with the students that just graduated, and it sort of illustrates what kind of calculation they are doing in this particular case. So we initiate trajectory at the interface in here, and we run them only up to the point in which we hit another interface of what we call milestone, then we stop. We stop and we record where they hit, and we record the time that it took them to hit the other milestone. Here we start the trajectory, we repeat the same thing for all milestones. We start the trajectory at this interface, and we run it until they hit for the first time another milestone, terminate them, and then record the data. The point is that these trajectories are indeed very short. If you start, for example, here at the top of the valley, you have a few trajectories going to the left, a few to the right, but they are at the end in the relatively small numbers of steps. What we collect from this calculation are essentially two functions of two entities. One, which is sort of the notative Kj, is the probability that given that you start from an interface I, you will end up on interface J. So you have, for example, in this case, I have four trajectories that I started at milestone or interface I. Three of them make it to J, one make it to K, so the total number of trajectories that I start is 4. The number that make it from I to J is 3. So I can give you an estimate of Kj in this particular case, which is 3 over 4, and of course I repeat it for all the milestones by this short trajectory. So this is one function, very important metrics essentially, that do the transition between all these interfaces of all the milestones. The second important function that I compute is the lifetime of the milestone, or it is the average time that a milestone, a trajectory that initiate at the milestone. So if I start trajectory from I, it can end up in J, or it can end up in K. I sum up the time and three combinations of all the trajectories and divide by the number of trajectories. Very simple. It's the average trajectory lifetime of a trajectory. These two functions are sufficient for me, if I do it for all repeatedly for all the milestones, to compute all the kinetic and the thermodynamic of the system. So I have K. K is the matrix of the transition that I just described, going from all I to J. This matrix is a transition matrix, probability matrix. It has some, it's not symmetric, but you can still have left and right eigenvector. The left eigenvector is an eigenvalue 1. Is the stationary flux, is the number of trajectory that under stationary condition pass interface, all these interfaces is Q is the vector for every element of the vector. If I find Q, I find the stationary flux of the trajectory. The problem, the computational problem is that the K can be a huge matrix because it depends not only on the interface but also the position in which I hit the interface. So typically what we are doing computed by iteration. We're having a guess. This is a guess which is just assuming that the system is close to a Q, and we call it Q0. If you set up the distributions of points, for example, sample at initial conditions for the trajectory, sample from a Coulomb distribution, and then we run a trajectory. Here we have some equilibrium distribution of points, which are sampled with canonical weight, and they are sampled in such a way that they are constrained to be in the mark. So we run the trajectory and we run it until termination. The endpoint of the trajectory are used in order to enrich the distribution and compute the second iteration. So the next time I'm going to run I will have new points that are coming in here that I can use in order to, in some sense, to continue those trajectories and see where they are going to end again. It can be shown that these things is converged absolutely, and whenever Qn is equal to Qn plus 1, the calculation for the flux are done, and then the result. In practice, however, in many cases, it's sufficient to use it in only this approximation, and I'll show an example of this type. So this is an example of the system that I've seen, that I just shown before. This is in the plane of the milestone. So we have the reaction coordinate sort of going into the wall, and this is the cross section that the trajectory is progressing. And what I'm comparing here is the different distributions inside the milestone, or inside the line that form the milestone. And there are three distributions here. The dotted line is just canonical distribution. This is the zero-order distribution that I just shown. The purple line is the exact distribution computed independently by long trajectory. And finally, doing spiral iteration, we are getting this kind of a blue line. They are coming out to be perfectly in here, even in one iteration, we don't really need to go back. They are doing option quite well also for this particular milestone with just the initial direct distribution. Here, close to the hole, we are deviating somewhat from we are deviating from the canonical distribution, but after a few iterations, we compare. We can show that it's actually quite easy, and with very few iterations, we're getting the exact value of the flux that are in the milestone. Now, so we know K, we compute this K, the stationary flux. What do we do next? So we have essentially now three entities. We have the K that I compute directly from the trajectory, the probability for going from milestone I to J. And I have also T, which is the lifetime of the particular milestone. So one interesting quantity is the probability that the last milestone that you cross is P, and this is given by the flux into through that particular milestone, the same Q that I just computed as the eigenvector of the matrix K, and multiplied by the lifetime of that milestone. So if you're sort of making intuitive sense, you're counting the flux that is coming in and ask how much it will live there, and that will give you the probability of being at that particular milestone. Then the two main observables, which we are interested in order to compare to the experiment, is the free energy, which is just the law of the speed that I have in here, and the mean first passage time given by the expression that is given in here, which is again the scalar product of Q at the lifetime divided by the flux to the last milestones at the product. In fact, in this short trajectory ensemble that we generate in this particular way, we can get information more than just the first mean first passage time. We can get higher moment of tau. It is information that is still buried in the short trajectory. We don't really have to go into any longer trajectory. We need more statistics because there are more complicated entities here. For example, Tij is the average length of going from particular I to J. But it is possible, and the equation becomes a bit more complicated, but it is quite straightforward. So, one more comment, which is related to theory, and bringing also back into Klaus, is saying the following. Okay, so now I have the stationary flux. I can also compute this K, this transition matrix, something that many people are calling the ultimate reaction coordinate, which the isochromator surfaces, the idea that was promoted by Pulholz, by Hammes-Schiffer and Attila, but the Peters and many other people. The chromator is a probability that I make it to the product for the first time before I make it to the reactor. And I can show that K, the same K that we have in computed the flux, if I now look for the eigenvector from the right, or I would the linear equation, so Kc, the commuter equals C, then the C is the value of the commuter for every particular mass. This is nice, and I can show one graph in here. I can calculate if these isochromator surfaces, this is for the Muller potential, you have a potential
energy minimum here and minimum there. We are putting a mesh in 2 dimensions and we can calculate the transition between all these points in the mesh. Then we calculate for C, and we color coded. This is, of course, if you're on this side, this is the product. You are going with almost mobility 1 to go here. When you're on the transition state, that is nonlinear functions now, it's not a plane, it's a sort of, it's a curve, consists of many states. The probability is about 0.5. The transition state is sort of what you expect. Half of them will fall back into the lateral state, and half of the trajectory will go forward into the product state. We can do larger system. This is a system of trifecta, it is water, even deep membrane. Just this is the plot I choose to show. Here is the line, of course, of coarser, because it's questions of statistics, but whenever you are there, you are committed to go to the product, that is almost 1, value to commit to 1, and this is the reactant of where I start, the value is close to 0, so here you are more likely to return to the reactant before you are making it finally into the product. And you can sort of draw schematically a line of maximum probability that will lead you from the reactant and here into the product, and it is going roughly that particular. So the committer is indeed a useful function, but there is an issue here that is a little bit frustrating for me. Why? Because I have 2 functions here that determine to be both the mean first passage time and the free energy. These are the stationary flux that is coming directly from K, and also the lifetime that is coming from T. So I need 2 functions that I need to compute, and K and T. And in fact, if I looked only on the condition the committer is completely determined by K only. There is no information buried in the committer in the so-called local diffusivity or the way in which the particles are moving between the speed in which the trajectories are moving between different interfaces. And here comes an idea that again is connected with Klaus, that maybe a better reaction coordinate, or at least additional reaction coordinate we should consider, rather than the either committer, is simply the mean first passage time. And you can see this is the closed equation for the mean first passage time. In this case, you have the K like before, but you also have on the right-hand side the local lifetime of the maximum, so you capture and you have an additional measure that is not being captured by the either committer alone. So here is the mean first passage time equation, and this is the paper by Klaus that essentially suggested that we use the mean first passage time as a reaction coordinate, something that we are just starting to do because we can compute it even for very large systems. For that particular simple system that I have shown, the complete MFPT looks very similar to the one that you get from the either committer, but we are working now on a more complex system in which we expect to see differences. This is expected, for example, when you have 2 channels, and one channel, the T, the local lifetime is very different from the other channel. So the decisions of which channel the system will prefer will depend also on the local mean first passage time. The trajectory marks only are very efficient, but I skip this as Sharon will be suspicious. So I'm going back to the example of anthrax. That is, we are interested in trying to understand the conditions of the N-terminal segment, which is all the structure, passing something that is called the phi clamp. This is a ring of regular rings that is sitting here. That once the N-terminal sort of passes and it gets stuck, it gets locked, and then it's sort of waiting for the rest of the protein to unfold, and then the conditions will occur. So the first thing we need is some idea of the reaction space, some idea of the reaction coordinates. In this case, it was not difficult to do, because just doing steering worked quite well. We generated a set of configuration which the N-terminal compresses until it passes the N-terminal in there and get hooked at the end. This is a set of configuration we can use as a simple representation of the reaction space. They don't even have to be on one line. Each one of them defines the so-called Voronoi cell, and then we have trajectory between the interface and the short trajectories of this type. So we collect K, we collect the T, and we are doing all the schemes that I just mentioned. For example, we can get a free energy. And the free energy that you are getting for this system, we can do it for a different population state. Remember, on the left side, when the protein is outside, the pH is low, and we can think about the 3 extreme cases, because we don't know exactly what is the formation state, but we are looking at an extreme case in which the whole things are protonated. These are the case in which none of the histone is protonated, and this is the case in which only histone is protonated, and this corresponds actually to pH equal 5, which is believed that this is probably the most closer to the Nevertheless, one of the leading curve here is of a high protonation state, which essentially is a downhill, and sort of support the notions or the idea that people suggest that this protein transporter is acting like a ratchet. What do we mean by ratchet? Is that a machine in which you're going only forward. You are really not allowed to go backward. Once you made it, you made the step, you're getting locked essentially in another equilibrium condition because it is extremely unlikely for you to go back. And that is essentially the case that we see here in which a system, rather diffusing significant part of the reaction coordinate, suddenly is falling down in quite a deep minimum here and make it very unlikely to come back. This is definitely not the case if the automatic numbers of protons being captured by the N-terminal is high, is low, So in this case, you have a high barrier and the system does not penetrate. Again, the low pH condition for transport of this protein is not necessary. And intermediate situation, you see the Schiessen limit is a clear minimum after we pass the 5 Planck, which is around 0. So once we pass the 5 Planck, this state is being locked down. The same feature is being captured also by the mean first passage time, which is sorted exponentially. And note that the sort of going in at a high, at a low pH case, in which is a high protonation state, is extremely fast. We are looking here at picosecond, and essentially the chain is penetrating inside the channel on the nanosecond timescale. But once it's inside the nanosecond timescale, getting out, once it is locked, taking millisecond, which is something that we're seeing here, which is extremely unlikely, and as I said, give enough time for the protein to unfold, the protein cannot simulate yet, and then being transported for the channel. Similarly, if you're in the wrong protonation state, if you are in a high pH state, then it's unlikely to get in, and if by chance you got in, you extremely quickly recoil back. The commuter function, which you can also calculate and think about, it shows very different behavior as we expect for high protonation state or low pH versus the other two states. In this case, the commuter function says that we are rapidly diffusing until half the way, or maybe a little bit more of the reaction coordinate, and from this point onward, we are completely committed, it shoots forward and it cannot go back, it's completely committed into the states of the product. The commitment to the reactant, if you protonate only the histidine or you don't protonate anything at all, then is much, much smaller and it comes only very, very late, which need to be forced. The question is, why this is so? What essentially drives the reaction in particular way? So the answer for that is actually very simple. Maybe we could have done this without doing this calculation. When you calculate the electric field of the channel, this is the right line, this is where the initial chain is terminated, you see that it's very negative. There's a lot of negative amino acids built into the span that is penetrating to memory. So all these negative charges is very attractive to highly protonated residues that are coming in now and passing the pipe down and getting locked inside the channel. So that is that. I don't know if I have any time anymore. Do I have any time? Right? I have time. Okay, so let me talk on another system that we can look at with this kind of approaches, this technology. This is the AJV reverse transcriptase, which is we did it in collaboration with the experimental group of Ken Johnson. This is a system that is a DNA polymerase for, again, another nasty thing, which is the AJV. And it is known to be quite selective. And it's a general question in polymerase, what determines the selectivity and what determines the rate? And we were focusing on this particular study is how the conformation transition of the protein can determine the rate. So if I just go back here. So there is the reaction having a number of steps. One step is the conformation transition. One step is the ligand coming and attaching the surface of the protein. Then there's a conformation transition, or lockdown, or reduced state, whatever you want to call it. in which the protein is closing on the substrate and the chemistry happens and something is released. We are looking now at what happened, or what is the difference in which we have a correct substrate attached to the surface of the protein, substrate that fits the DNA strand that it wants to be added to, and another one is a wrong substrate that is fitting it. We need to compute the action coordinate, and this reaction coordinate is computed with some functional technology that is in my book that I did not describe. The thing is done in water, when it's done if we are getting the free energy in this calculation as well. Let me just run this again. Because the overall change in the structure, you see the backbone is not very large, right? It is emphasized what is going on here is mostly looking here. Here's magnesium ion, and here is the base pair that want to be added into the clean strand. We can get a somewhat closer look of what's happening in the active cycle. The reaction called the lysine group are closing on the base pair, and sort of take care of the charge of the nucleotide and make the system more ready for reaction. This is the free energy that you compute, and there are several steps that you see here. This is the point that you just rectangly attach to the surface. We match the two curves here. There is some differences also in the binding to the surface of the correct and incorrect substrate, but at the moment we do not consider them, so these two curves are just artificially matched together. And then the protein is undergoing conformation conditions. There's a barrier that needs to be passed, and then is the deep minimum one, the protein sort of locked around the substrate that is coming in here. We see here that if we are having the correct substrate, which is the red curve, the minimum here is very deep and similar in some way to the Russian model that I had before for anthrax. It is extremely unlikely that the protein will go back to the release even at this point. The barrier of climbing up in here is, 25 kPa more, never happened in the lifetime. However, if we are bringing in incorrect substrate, which is the blue curve that you see here, the minimum of the blue here is actually higher than of the free substrate. So once the substrate or incorrect substrate succeeds to come in, and even if it is temporarily trapped, it will very quickly be ejected, it's thermodynamically unstable, and there's going to be another chance for the protein to try to bring correct nucleotide in. So, and once the substrate is here, it can continue here to do chemistry, and essentially it's fully committed like a ratchet of continuing into the chemical reaction. This is the time scale, which is sort of shown in nanoseconds, but you're seeing that you're going up to 10 to the power of 8 nanoseconds, so we are in a time scale that is really pretty long. This is experiment versus computation. So for the incorrect substrate, which is the TTP in here, we are seeing that the K, this is the incorrect substrate is here, the ATP, and see that the probability of the data going back is very high, it's comparable to whatever going in. Like the correct substrate, the big difference as far as the information chain is concerned, is going backward, which is K minus 2, which is 4, very, very small, only 4 times in a second, versus 2,500 seconds, which is going forward. And that's essentially a selectivity mechanism of picking up only the correct substrate. So that's the conclusion, and thank you, Klaus, and thank you, audience, and thank you very much.
Well, we're only 15 minutes behind schedule. So, a question?
Now you say another full 5 minutes.
I think we So our next speaker is Professor Ivan Beckman and of the University of Illinois, and he's going to talk about capturing biological membranes and membrane proteins in action using advanced simulation techniques.
Okay, well, the last speaker, so even though I don't have much time, I decided actually to include quite a few slides about Klaus, because that's what he suggested, and if I don't have enough time to talk about the science, that doesn't matter, so because we can chat about it later. So I decided actually to include a few slides just to talk about Klaus's impact and legacy, some of the aspects of his impact and legacy, which I think are important. You heard a lot about his impact as a scientist, as a teacher. So I thought actually, again, being part of the center, it's important to realize that he really, as pointed out by several previous speakers, he was actually the person who developed tools, and he made an effort to make those tools available, training people, telling them, showing them how to use it, etc., etc. And these are some statistics on the two flagship programs of the center that he started. So by very conservative measures, we have about more than 100,000 users of EMD. If we counted a little bit more relaxed, about 200,000. And in the other program, between 20 to 45,000 users, and many of these are NIH funded. And as John pointed out earlier this morning, if you don't believe these numbers, just look at the citations that these programs receive. So collectively together, every 2 hours, these programs are cited. That means people are using them. Many of them, of course, don't publish papers. Many of them don't even cite them. But those people who do that, every 2 hours they get one citation. So this is what I would seriously consider the high impact of what he started. So he has a lot of material on the website. You can see some statistics, really impressive number of people around the world are using the materials that he worked so hard. I know Zan knows it too, how much he was caring about the website and the material on the website. Every group meeting we had comments from him, this should be fixed, this should be added, etc., etc. So this is in terms of the website. You can see again a little bit more details. These are actually statistics over 5 years only. And you can see it's really impressive number of publications, how many downloads. In particular, tutorials and training materials was something that he concentrated on over the last 10 years or so. And so I was very proud of the number of hands-on workshops that he sort of gave over many, many years, starting in 2003, where we had the first one here. More than 100 people came here. And I can tell you, and many group members are here, this wasn't easy to develop all the materials for that workshop. Everybody remembers the month that people worked on those materials. And again, more than 600 people were trained through these workshops. And again, almost 2,000 pages of materials are available on the website. So he was very proud of this, and we did a lot of workshops. And I can tell you, after a while doing them, I think many faculty members in the center were participating in, including also staff members. But I and him were sort of the fixed part for a long time, 2 days Klaus, 2 days me. So I traveled a lot with Klaus, and at some point it becomes actually a burden, going all over the place and, staying a whole week and doing all sorts of things. But one thing that actually really helped was Klaus was really a fun person to travel with. And I think, again, you can see some examples here. This is the first workshop we had in Frankfurt, Biophysics workshop in Frankfurt. So we can see hands-on workshop, computational biophysics. And he always made an effort to make it fun for people who were traveling with him. So you can see we are in a beer garden here with Zan and JC and two other students here. I'm sitting on the other side taking this picture. And I have my own picture here. And you can see around, actually, at that point, he arranged a travel sort of trip around the Rhine River. So, just went along the Rhine River. So you can see we are posing to shoot these cannons when we were visiting the castle here. This is another example. He was a great fan of seafood, as many of you probably know. This is the League of Seafood in Boston, that again, another workshop. And you can see we had fun. That was one of my first big lobsters here. So we had a lot of fun moments, and this one is actually the one in San Francisco, the workshop we had in San Francisco again. You can see a sort of bike trip was arranged by Klaus and Zan, and all the students are taken to nice places. So Elizabeth might remember these photographs because I'm covering. Actually, I got my green card on that day. So, and funny, I had everything green on me. So you can see green shirt, the green shorts, and everything. Sorry?
We drank margaritas.
Yes, and we drank margaritas here, which are green, by the way. So this was really a fun moment. But I think actually that's really showed something about Klaus, that he really cared about his students and postdocs. I witnessed many occasions that he was trying to protect them against the system, administrators, other staff members trying to take advantage of them. And every time he took them to trips of this sort, he tried to make it fun for them. It's not just taking them, making them work. So he really did his best to make it fun for the students. It is also actually in a van every now and then getting together. So I came here in 1998, so it's a long story. I can tell you how I managed to invite myself here, and then we spent some time here, so you might recognize some. This is a very old picture. Some of us are in the audience right now. That's me back there. So 4 weeks I was here, and they generated enough material to put the paper together with Sharon. Yeah. Another paper later when I visited 99, and then I joined actually back then. Zan and Klaus took me to a sushi place, to Co-Fusion, which used to be on university. And I almost killed myself because I thought wasabi was just some sort of vegetable. So they were kind enough not to say anything about that. So anyway, I don't have a picture of that. This is another interesting picture of a colleague, Shigei Hayashi, who was working on bacterial adoption with Klaus and me. Every time somebody left, they would receive this surprise gift from the group, which was essentially a university book. And then, so he was shorter than us, and he's trying to actually make it as long as us. That's a funny picture. We published a lot of papers together with Shay. And actually, I'm proud. Zan, said he can't beat you, of course. You were really the closest collaborator, and Attila, you were the longest time collaborator. But in terms of publications, I think nobody can beat this number. I have 42 joint publications with Klaus. He is my number one. I am his number one co-author, and he is my number one co-author. So very proud of this. So we have had papers all the way when I was student visiting, and then throughout my sort of independent career. And I think, some of them, this was a science paper on aquaporins that we published together. It was fun. And then, related to that, we got this sort of award-winning image in 2004. This is again aquaporin showing water molecules going through the channel. And as I always say, this is the closest I get to the Nobel Prize. And so this video that was put on the Nobel Prize website in connection to Peter Albrecht and his Nobel Prize in chemistry. All right, so let me now tell you a little bit about the science. So now I was working on channels when I was with Klaus, and then I became independent, staying on the same campus. And I had to stay out of his big shadow, which was covering the entire country and the world. So I switched to transporters, and that's actually what I've been working on since then. And so these are important gatekeepers in transporter material across the membranes everywhere in the body. So every time anything in the GI, in the kidneys, you take any drugs, it has to be transported across many, many membranes to get to the site of action. So you have these molecules, these molecular machines involved in the process. So these are major sites of drug action. So you very well-known antidepressants, for example, act on neurotransmitter transporters. And these remain to be actually major target for drug development. And they also control pharmacokinetic of drugs. Some of these pump, for example, drugs outside the cell, developing multi-drug resistance. So one thing that is important about their function is they undergo very large conformational changes because they have to follow the so-called alternating axis model. They can't be exposed to both sides of the membrane because otherwise they would be acting as a channel, not as an active transporter, which is able to pump stuff against the electrochemical gradient. So due to this, they are very slow, and they sort of go through many, many conformational intermediates and states, major functional states. And that's what people are trying to actually characterize using experimental methodology, trap them into different states and get this structure. So I show you actually what we have been doing in terms of getting these structural transitions using some of the methodology, and in particular I would like to point out how much the lipids environment matters, and that goes along with sort of what Klaus insisted, that we really have to describe the system as detailed as possible. Really, in biology, every little detail makes a difference. So, just to point out, so again, I mean, you might see different transporters, and again, I don't get into the details, but they come in different flavors: sym-port, anti-port, uniport, and even though the conformations that you're seeing here are very similar in terms of the flip-flop motion that they undergo, the coupling of these conformational changes to the presence of the substrate in the binding site is extremely important in terms of determining whether there's a sym-porter or an anti-porter, which have completely different physiology. The same protein architecture, one of them is supposed to flip in an empty form and is forbidden to flip in its partially bound form, whereas the other one is forbidden to flip in an empty form, and it can only flip when it's singly bound. Again, this really determines the mode of action, and it's amazing how, by changing a little bit in the protein architecture in the binding site, you can control this behavior. So we are really interested in the chemomechanical coupling. What happens when the substrate binds, and how it affects the free energy landscape that controls protein conformational changes from one state to another. So that's the theme that I think we have made some progress, and I'm showing you some. So the alternating access model comes in different flavors. Again, they have to be accessible from one side of the membrane at a given time. And this comes in different forms depending on the source of the energy, protein architecture. You can see 3 examples shown here. And these are, of course, very, very slow for atomistic simulations. There is no way we can do these millisecond or much, much slower processes, as you heard also from previous talks. So we have to sort of apply biases. You have to push the system to walk along a particular pathway. And this pathway, this keyword here, is extremely important. What pathway you're going to push it through. Because these are much more complex than some of the examples we heard earlier, so it's not really clear what is the reaction coordinate along which you want to bias the motion of the system. So, and of course, I mean, eventually we would like to be able to, when we use this transition, to describe the free energy landscape around this pathway or governing the motion of these transporters. So really the key for us, and for many, of course, one of the key aspects is to define the reaction coordinate along which to study these systems. And of course, one complication and one thing that we are realizing is important is the presence of these lipids. So you would like to see this in the presence of these lipids. This beautiful picture is, by the way, made by BMD, and Chris Main, who's in the audience, actually made this beautiful figure. Klaus got a little jealous when he saw that, and he immediately published that as a cover page, actually. Similar, something like that. So it's beautiful sort of picture showing the membrane. Yeah, so that's what we would like. We would like to look at the motion of this protein in the presence of the lipid, and you can see how intimate the lipids and proteins are. So really, if you want to understand how it works in the membrane, we have to include these lipid molecules sort of explicitly in our simulations. So that's the problem. We have an unknown landscape. You want to go from telluride, like this valley, to the other one. And you can take and actually sort of simplify those sort of simple initial guess and do some methodology to define this pathway. But if the actual transition pathway is far from the initial guess, then probably you won't have a chance to reach there, even if you apply very sophisticated sort of path refinement algorithms. So we have developed, or we have taken a different approach in which we are actually searching the space a little bit more thoroughly. We spend a little bit more time exploring the configuration space. By no means is it going to be perfect and complete, but at least it allows us to maybe find better reaction coordinates and pathways. So in this representation, so we're going to actually spend a little bit more time instead of just working with RMSD, going linearly from one point to the other. We're going to explore the space based on the protein architecture, number of helices, etc., etc. We're going to actually explore the system and then compare these initial pathways by based on the non-equilibrium work that is required to walk the system along this pathway, again, as a semi-quantitative measure. And this way, actually, they have been able to sort of take a better initial test to start with. So this means that maybe here we are using this biasing protocol instead of just RMSD. We have a variety of corrective variables implemented in NAMD. Actually, they're all available. There is subgyrration, rotation, quaternium, etc., and we can combine them and use them in different linear combinations in different orders, etc., etc. So that has been actually the approach we have been using. So it takes some time initially, like for example, in the case of this drug exporter that goes from this state to the other state, if you look at this, we are looking at 200 independent initial sort of exploration of the system to see how to go from A to B, and they take, of course, different pathways, and we compare the work, and then we decide what might actually be the easiest pathway for the protein to walk from this corner of the room to the other side. So now we have actually, so again, I mean, this is all done in the presence of the membrane, so this is a pretty realistic representation of the system. And now that we have this, instead of working with one snapshot from the crystal structure, we are able to look at many, many conformational states. For example, if you want to do drug docking or inhibitor docking for this system, instead of just working with one crystal structure, one snapshot, we can take, let's say, 50 snapshots or 50 points along this transition, and then use them, all of these for our docking studies. And as you can see in this particular example, this is one of the crystal structures. As we go and explore the other state, they have a better chance to find high score drug binding to those pulses. So that increases the scope of your sort of rational drug design approach because you have multiple conformations to work. So this is another example. That was just a protein to see how we might describe protein motion. So now the challenge, as I pointed out, is how this large-scale conformational change of a protein is affected by binding of a little substrate inside the lumen. So we went to an antiporter. So this protein, this particular protein, is supposed to flip only if it's bound to the substrate, and it should not be doing this transition when it's empty. So we set up this thermodynamic cycle, so we're going to look at all these four legs using these computational techniques and advanced sampling methods, and see whether we can actually describe this difference in free energy barrier for these two transitions. Okay. So now, as you can see, actually, that's something that experimentally is proposed based on sort of very schematic description. In the apo form, there has to be a high free energy barrier. Once the substrate binds, you form a new state here. For the left state, you form a new state here. For the right state, and the barrier separating that should be lower compared to the apo state. But of course, this is a schematic. There's no number here. This is how it should work in principle. So we have done again our approach to this to determine sort of what would be a good pathway to induce the transition, working with the individual helices, changing their orientation, trying different pairs of helices and see which way the protein is more comfortable to do this transition. And then use this to construct a sort of large vertical exchange. We have a sampling simulation in which we are really going to look at the entire cycle in one big simulation. And actually, that's what you get from that simulation. You have 150 replicas going through the bound transition, which is shown in green here, and then going back to the unbound, bound, or apo form, which is shown in red. And as you can see, it exactly replicates what people suggested should be the case. But again, here we have all the details. This is a protein, fully atomic representation inside the membrane for which we are doing this. So we seem to be sensitive to the presence of the substrate. We can understand how substrate binding affects this transition, reduces the energy barrier, etc., etc. So now it's still expensive. It took us 1.5 years of kind of calculations, but we seem to be able to now pick up the chemistry and maybe look at mutations and find out what happens due to different mutations, etc., etc. So let me skip this one. and then show you just one more example of a neurotransmitter transporter in which we are, again, using a similar strategy. So this transporter forms a trimer. Each monomer works independently. And if you simplify the representation of the transporter, you can have a sort of a stationary part of the transporter known as the scaffold that actually contributes to the formation of the scaffold in the middle, to the trimerization domain, so to say. And then you have the transport domain, which moves in an elevator type kind of motion. And that way, you can change the accessibility of the substrate binding site from one side of the membrane to the other side. So this is essentially what happens if you just look at one of these monomers. So it changes its orientation and position with respect to the scaffold domain. So we selected those as a natural kind of space that we would like to explore, because nobody knows whether it moves and then rotates, or it rotates and moves, or is it stepwise going back and forth. So we really don't know. We just know the end states, inward facing and outward facing to this program. So this is essentially the space we defined, translation versus orientation, and then we're gonna try to sample this space using different, steered MD initial, just to see this space with some initial confirmations. We know some of them from crystallography. These are some trapped intermediate states, but we would like to cover the entire space to be able actually to see how the whole landscape looks like. So you can use different tricks to do this. And again, we are going to look at 3 bound states. A fully bound state should be able to flip. The empty state should be able to flip. Fully bound state should be able to flip in order to go around the cycle, but a partially bound state where you have only one of the ions or one of the substrates should not be able to complete this transition. So we're going to look at 3 different states and see if we can actually capture the chemical dependence. So these are some examples how we are going to actually sort of explore this 2-dimensional space. We can use different assembly protocols, move along translation, rotation at the same time, move up then to the left, and then up. And we try to cover as much as we can this 2-dimensional space using some assembly protocols. Then actually we have covered pretty much actually a large fraction of this area that we would like to cover to be able to get the free energies. It's not enough. We sort of define a grid, and again we select some of these points to fill these gaps, to seed them further. And by doing this, we are, at some point, we are able actually to cover pretty much the space. That gives us an idea about so we do actually, again, an approximate free energy landscape at this point using 2-dimensional bias exchange umbrella sampling simulation. That gives you an idea about the shape of the landscape, so suggesting that probably we shouldn't worry about these high regions on the top right and the bottom left. We should maybe focus a little bit more in this region. That is great, and then we can actually set up another round of sampling of the most important region that we are really interested in. By going through this, actually we can get a better estimate of the free energy and use the free energy path, but and then sort of by post hoc string method or string method with sorts of trajectories that you heard from Benoit, you can actually further refine this pathway. So once we are done and we have some idea about this path, then we take one-dimensional path and do one-dimensional vertical change over the sampling on that path to calculate the free energy along that particular path. So, and again, as I pointed out, we are going to look at the fully bound state, which is supposed to flip. APO is supposed to flip, and then the partially bound that is forbidden. It should not be flipping, otherwise it produces a leak and you lose your ion gradient. So this is how much time went into this very expensive calculation, 150 million hours of supercomputing units from our InSight grant. So that's the free energy that you get. So let me compare. This is the fully bound state. You nicely go through a barrier. Our intermediate is different from the crystal structure. We explained that by the fact that the crystal structure is actually trapped. It's a cross-linked structure. outward facing is similar, inward facing state is also different. But again, I point out these are micelle cellularized proteins, and there is no reason for them to match our minimum energy state, which is done inside the membrane. If you look at the APO again, we have a slightly higher barrier here, but still the transition is possible. The position of the minimum energy states are different from the fully bound state, and that should be because it's a very different state. And if you look at the partially bound, which is not supposed to flip, we don't even come back down starting from this corner because something is preventing the transition from taking place. It's difficult for the system, which is consistent with the mechanism, and we haven't quite understood why. So we are looking at these details and see what kind of chemistry, what kind of interactions. We have some ideas about the position of some of the loops that might be in the way, preventing the flipping from taking place. We haven't quite understood this. But again, so that this is exactly what you sort of expected. APO and fully bound states are able to complete the transition. There is a barrier, and these are slow transitions anyway, whereas the partially bound state doesn't seem to be able to complete the transition in our hands. And that is actually make you again look at take one of these paths, the lowest energy path along this map, and plot them like this. You can see that the APO has a slightly higher energy barrier compared to fully bound, which also agrees with the thermodynamic cycle measured experimentally for these states. So what we think is happening is, again, we have a sort of going through stages of translation, orientation change. It's not quite linear, it doesn't go all the way from one side and then the other one. So it's a combination of translation and orientation that completes this changing the accessibility of the substrate from one side to the other side. So let me skip this. Lipids are extremely important. What I showed you was the average behavior for the 3 monomers in one simulation. But again, if you see how lipids are closely interacting with the proteins, and in our limited simulation, we don't have enough time to symmetrize these interactions, we can expect actually different behaviors for the 3 monomers. And that's actually exactly what we see. So you can see actually as you go, move along this, there are different parts of the protein that are interacting with lipids, and that is of course affecting the behavior of the 3 individual monomers, resulting in some variance in the behavior of different monomers as expected. So lipids are playing a really important role. As you can see in this example, they are closely interacting with the protein. So again, another question was, what part moves inside the membrane? Is the green part going up, or is it the orange part coming down? But as you can see now that we have done this simulation in the presence of explicit lipids, we can say exactly what happens. Initially, one of them moves up a little bit, but then they move down together, etc., etc. That was also another question that people have in the field. But actually, let me continue with a couple of other examples in which we are looking at how closely lipids are interacting with proteins and how much, from a functional perspective, they participate in the function of protein. This is another example in which our colleague Eric will solve the structure of this channel. This is supposed to be an open channel, but if you look at from the bottom, it's closed, and from the top, it's closed. In the absence of lipids, you can't even explain how can this be a channel going across the membrane, allowing the ions to go across the membrane. So only in the presence of lipids, when you place them, you can see that actually the lipids in this half, in this leaflet, are actually covering or participating in formation of the ion permeation pathway. You might actually notice some of the lipid head groups down here, right in the middle of the membrane. So that's just one example of how closely and how actively they participate in the function of membrane proteins. This is another example in which actually lipids are transported by a protein. You can see how a lipid is using a protein track. This is a scramblase that scrambles the lipid composition of a lipid bilayer for you. This is a front view and side view of the same simulation, and you can see the position of the head group of the lipid as it goes from one side to the other side, this is something that we captured in a microsecond simulation. So this is another example. This protein is, by the way, calcium activated, and if you remove the calcium, then it's not active, and it shows actually what happens to the protein structure. But the main point is really lipids make a huge difference. We know that experimentally also. We are trying to move away as much as we can from micelles and other solubilizing agents and put the system in a natural environment. But it's just the beginning of discovering actually their role and how many ways they can participate in the function of membrane proteins. So I spent actually 5 minutes. I'm on 28 minutes. So I spent 5 minutes just to tell you a little bit about some new direction in my group, which was sort of inspired by Klaus's insisting that we have to go to the cellular level. So that was really his dream, and he made huge steps in that direction. And again, my group within the center is working mostly on membranes, and I would like to just give you a couple of examples of what we have been doing there in case there are opportunities to interact further. So one area is actually we would like to have automated methods to allow you to put together sort of cellular envelope, just the membrane of the cell. And these are some of the steps that we have been applying for a simplified spherical membrane for now, but I will show you membranes of different shapes. So we have algorithms that allows you to nicely pack the exact number of lipid molecules that you need. Based on the Fibonacci algorithm, you can actually pack them here. Then you can take your membrane protein, and you might have hundreds or thousands copies of these membrane proteins. So super coarse grain then in a way that the soluble part and the membrane part are distinguished, and use this heavily coarse grain system. We have algorithms to do this automatically. Using this now, you can actually throw in your many proteins, let's say 100 copies available from experiment. You can throw them in and use a sort of a gravitational field coming from the position of the membrane to initially distribute these proteins. And then we can use this to place the real membrane protein, create densities corresponding to these proteins, and use those to carve out exactly the shape of that membrane protein that you need to be able to place the protein perfectly. It sort of snugly fits into that position. Because when you go to this scale, a lot of challenges, new challenges you have to face with. If you have a little bit of a gap for every little protein, and you have, I don't know, 2,000 copies of the protein, your simulation is going to crash because you're going to collapse this process. So this is what this result. This is one model that we generated. So we can see we are trying to actually include as much as we can experimentally known features in this. This is the information that Roux collected. So based on the minimal cell approach, this is just an envelope. It's not the cell. It's just an envelope. But we are also actually including the based on the you see here we have about 1,400 membrane proteins included here. So this is about 113 million Martini particles, or about a billion atom system, and we are trying to actually push this to get the simulation going on for supercomputing 2017. So this is another example of the endoplasmic reticulum. This is much larger. For this guy, we do have an electron density map, and we are using this density. The challenge is, if I give you this shape, are you going to be able to build an atomistic level model of lipids inside this? And what you are looking at actually is an atomistic model of this beast. This is 1.6 micron large. The EM map was done experimentally here. We couldn't even actually load this into EMD. Noah, who did this work, had to do some tricks. If you're interested, we can share with you to be able just to do this animation. You can see actually the two leaflets of the membrane. This is the lipid composition we are using. Again, try to get this from experimental known feature. You can see the two leaflets here. The black guys are cholesterol shown here. And he has a sort of a set of algorithms known as XMass builder, or experimentally derived membrane of arbitrary shape. Any shape you give us, we can create this. And I can tell you there are many challenges when you deal with these sort of systems. So just generating the file for this thing sometimes takes hours to learn that it wasn't working. So this is another actual example, and this is the last thing I'm going to show you, in which we are trying to change the shape of the membrane in an arbitrary manner. Whatever you like, this is the work of this name. So we can actually create the shape that we would like to have, and then this could be your favorite CAD program, Blender, or whatever, and then you convert this to some densities that we use in combining them with MD simulation. What you're looking at is a coarse grain level simulation in which we are in a 10 nanosecond we are using this shape. He has done this also atomicity. It's a little bit slower, of course, but we can get it right. So we have the right protocol to use these shapes. You can somehow compare this to the beginning of SND, which apply the force to induce a particular change. Now we would like to do this completely independent of any corrective variable. So I want to go from shape A to shape B. And of course, the pathway, the mechanism, is it right or not, the energetic, these are all coming. And then this is, here is changing the density that controls this additional potential is changed in a time-dependent manner. And you can see we can actually induce a process here. So this is a nice sum function that can be done periodically. And the final example, and I'm going to do this very fast because we don't have time. If you're interested, we can talk offline. The final example is now, again, through a nice combination of a plane and a sphere and then intersection, then he is able actually to design a mechanism, and this is just a prototype for a very small system, in which we are inducing the formation of a vesicle in a molecular simulation. And of course, the reverse of this would be the fusion of this, and now we'd like to put proteins here and see how the presence, let's say, of snare proteins or other proteins might facilitate this process. So we are very excited about this. So I'm going to skip the acknowledgement, just because I acknowledge a lot of people, a lot of computer time is really essential for what we do, and thank you very much for your attention.
Any questions?
It's more a request for comments. So what do you think about something? In your mapping of the conformational states that you express array proteins that does, you're using essentially normal dynamic simulations that you guide through different emission paths, right?
Not long, short. We do another 10, 50, 20 nanosecond simulation. Just quickly over here, there. And because these are very, very fast, a lot of hysteresis. But again, if you do many of them, you start seeing that some of them bundle here, some of them bundle up there. It seems that this guy is easier than the other one. So that allows you to explore it.
But you're going always from point A to point B. You're trying to go entirely.
Right.
There, as you know, the people are using Microscope models and try to map all the conformations to try to see how connected they are dynamically in the energy landscape. Is it something of, can you compare, does it make sense to compare this to approach?
I think actually what we discussed with Vijay and actually Diwakar, who is here, so is to maybe combine these two. Again, in those Markov state models, still you have to slowly walk and sample, and sometimes you don't go too far. So if you can combine this with those Markov states, so that might be actually a good, effective combination. See that.
All right, let's thank our speaker again. And our final speaker is Ted Brown, who is the founding director of the Beckman Institute.
This will be quite a day. I'm really very happy to have the chance to put the closing remarks on this day, and I appreciate being invited to do that. I think the best way that I can honor Klaus's memory is by perhaps saying a little bit about the history of the Beckman Institute, and because Klaus really was one of the originals, And I'm sure that many of you are too young to have been around to know how all of this began. As you probably know, I was the founding director of the Beckman Institute, and I was sort of an overseer of the proposal that was submitted to the Beckmans to build the Institute. And the Beckmans were extraordinarily generous. They gave us a lot of money to build this big building, and it was understood that it would embrace an unprecedented range of disciplines. This institute was, at that time, I think maybe still is, the most broadly based interdisciplinary institute in academia. So we had cut out for ourselves quite a challenge. And so the first thing we had to think about was who's going to be in this place? I mean, we did have some ideas, but we had a very wide range of options to choose from and inviting people to come into the Beckman Institute. And so we didn't really actually have an algorithm that you could apply to this problem. It was, in a way, it was sort of, we were having an embarrassment of free ranges of choice. And so we had, this great proposal for an interdisciplinary institute, but how do we put that into practice? How do we actually make these choices? And so we had to think about who would be in here, how they would fit together, and so we were kind of feeling our way. And I should say that I wasn't alone in this enterprise. At this point, Professor Carl Hess from the Department of Electrical and Computer Engineering and a professor of physics was a co-conspirator, as it were, and Bill Greeno from the Department of Psychology also. So it was sort of the three of us who, in the end, we really were the ones who had to make the choices about who came into the Institute and in what measure. And so we were kind of feeling our way. And Carl Hess had done postdoctoral work with John Bardeen. And he went to see John, and John had been involved a little bit in the discussions with Beckman Institute. So he went to John and asked him, how would you make the choices? What kind of criteria would you use in making the choices of groups or individuals to come into the Beckman Institute? And John Bardeen was sort of a demigod of science around here. He had 2 Nobel Prizes. He was a very quiet man. He didn't say anything very quickly. He sat there and looked at Carl for a while. And he said, group your interdisciplinary research around powerful ideas and machinery of the disciplines. And that actually encapsulated just about all the decision-making we had to make. And I think we could take that to heart. And certainly, Klaus Schulten was one of our signal successes in applying that formula. When you think about Klaus and the kind of ideas that he had and the focus he had on creating the machinery of his discipline, it was a wonderful choice. So he was recruited to come here to Illinois from Germany on the strength of his being able to establish a group in the Beckman Institute. I think that was part of the negotiation, wasn't it, Zan? And Zan, of course, came with him. And so he was going to be in this group. Going back to 1989, when the Computational, when the Institute opened up, and Klaus came in at that time as we began bringing groups into the building. At that time, computational capacities were small in comparison with what we have today, and Klaus was all about computation. So, as you know, the aim was to create this tool such as the computational microscope. He was determined that it wasn't going to be just a matter of a look and see thing where you built this model and look how nicely it conformed with experiment. He wanted to build something that was so good that it was actually the thing that you could use to make predictions about what might happen. It became a tool for scientific advance. You could really use it as a tool in scientific methodology. So, it would be, I guess, a sort of an understatement to say that Klaus was energetic. And maybe demanding would be a good word. After we got settled in here, he came to my office door fairly regularly. And he came in, always with that lovely smile and then blinking his eyes with enthusiasm. I'd tell you about this and tell you about that. And oh, by the way, I need a new workstation. Oh, I need that wall moved up there. there. I need, you know, money for matching grants and things like that. There was always something. Carl came to the door. Carl came to the door quite regularly. Well, we were living in a pretty sort of, I would say, fevered atmosphere around here in those days. You know, the National Center for Supercomputing Applications was in this building. It had its headquarters up on the 5th floor. And it was, of course, it brought with it a great number of computational tools. And there would be connection machine and this machine and that. Although the major Cray machines were over someplace else, but Klaus was in close proximity to a lot of those computational tools, and he took every advantage of it. He had that army of guys who really knew how to make the best of these things. One in particular, I remember, Larry Smarr and Donald Cox, who was a visualization person, got the attention of Jim Clark, who was the CEO and founder of Silicon Graphics. And Clark gave the Institute 20 state-of-the-art Silicon Graphics workstations, which were put into a room on the 3rd floor up there. And so it was kind of a matter of who's going to, NCSA had the control of it, but it didn't take long for Klaus's group to figure out how to squeeze the blood out of those machines and you know, run in parallel and do all the things that they had to do. So I think in a way the Symposium, in its part, when you listen to all the things that were said, it's sort of a history of how Klaus managed to leverage and parlay his understanding of computational, not just the computational algorithms and so forth, but his understanding of computing as a machinery, you know, and how to make it work. So I really think that when you look back at what John Bardeen told us, it was Klaus really, and it was a wonderful example of somebody who knew how to build the machinery of his discipline. Sam and Klaus together were a wonderful couple, and she's of course a chemist, and I'm a chemist, so we had things to talk about. We even talked about general chemistry once in a while. And I remember one time in particular when Sam and Klaus, we had them over for dinner, and they appeared at our house with a tray. I don't know if you remember this. Yeah, but they gave us this wonderful tray, which had on it it was a copy of a map from some ancient document, not so ancient, perhaps maybe the Renaissance period, but it was in French. It was a diagram of a terrain, magical terrain, and there were little houses on it, and the names were all in French. So my French is not one of my languages, so I had a little trouble wondering what some of those little houses represented. But some of you can sort of figure out. And that tray, by the way, is, you know, we've never lost that tray. It's in our home. We use it all the time. We think about you every time we use it. So I guess I want to say at the end that you know, when Klaus came to the Institute, it wasn't immediately obvious that he was going to be successful, not to the degree that he wanted to be, or that we could imagine to be, or to the degree that he actually turned out to be successful. But, so in a way, he surprised us, but I think of it as, you know, that's the sort of thing that heroes do. They just surprise you, and they inspire you. For me, Klaus is a sort of enthralling figure. You know, he was one of a kind, and he was a friend, and I'm going to miss him terribly.